So, Kranti madam.
Yes, sir, we can start.
Yes. Professor Churungu is already there.
Yes,
so we can welcome him and we can start. Please, it's over to you.
Yes, sir. So just a minute. Just a minute. Just a minute. Good morning, everyone.
Good morning.
On behalf of Bionavid and IPS, I would like to welcome all the dignitaries, speakers, and all the participants.
Your voice is make it a bit loud.
Yeah. Good morning, everyone. I would like to welcome, on behalf of Bionavid and IPS, I would like to welcome all the dignitaries, speakers, and all the participants to day 2 of the IPS Bionavid collaborative webinar titled Next Generation Sequencing for Deciphering Host Pathogen Interactions. After an eventful first day, we are back today with another knowledge-packed day for you all, featuring sessions from eminent scientists and academicians. We have with us Dr. Nikhil Trungu, Professor, Department of Botany, Northeastern Hill University, Shillong, and Dr. Binod Bihari Sahu from NIT Rourkela. Both of them will be presenting their take on the role of omics in general in understanding host pathogen interactions in plants. We also have a short session on an optimal NGS data analysis approach using one of Bionavid's most interesting and exciting products, that is the Genome Station. That session would be presented by yours truly. So we have quite an interesting day ahead of us, and I hope all of you would be looking forward to it. So without further ado, I would like to kickstart today's proceedings by introducing our first speaker of the day, Dr. Nikhil Trungu. For millions of years, humans have used plants for food, raw material, and medicine. Since the utility of classical molecular genetics beyond model organisms is very limited, the vast specialized metabolic systems present in Earth's flora remain unutilized. Professor Tungu, who is currently working as Professor, Department of Botany, Northeastern Hill University, Shillong, has over 33 years of teaching and research experience with more than 50 publications to his credit. Apart from his usual academic and research duties, Dr. Trungo is serving as the president of International Buckwheat Research Society, along with being an active member of several national and international level committees and task forces. Through his talk titled Application of Integrated Omics Technologies in Deciphering Specialized Metabolic Pathways in Plants, we are sincerely looking forward to Dr. Trungu shedding more light on the above mentioned topic. So over to you, Dr. Trungu. A very warm welcome.
Good morning, everyone. Am I audible?
Yes.
Yes, sir. Yes, sir. You are.
Good. Primarily, I'm not a man from host pathogen interaction. So, whenever I was approached, I thought of what I should speak over here because this webinar is being organized in collaboration with Bionavid, and we did have some sort of collaboration with Bionavid earlier where we got certain things outsourced from them, And I thought of let me talk about something which we have done in collaboration with Bionovid. So I'll be taking a holistic view of the omics approach, and as we know over a period of time that when the research activities in the area of molecular biology started, people started looking at enzymes and the proteins, and the whole thing shifted to genomics, wherein the entire genomic DNA complement of an organism was studied. Associated with this came up the area of proteomics, where the proteins and the differentiation aspects of protein turnover, their activities, all that was studied. Other omics technologies came up, transcriptomics, and then metabolomics came up, and we had intractomics, we had degradome. So all these omes came up into the picture. The question is, did these all omes individually solve our problems which we are trying to understand, or was there any need to integrate all these technologies together and see whether we can get a much better output?
Dr. Sungu, I am interrupting
Yeah,
from IPS office. Can you make your slide presentation mode?
I have it in the slide presentation. Okay,
Yeah.
okay, I will.
Bottom panel.
Okay, is it okay now?
Right. Yes, correct. Fine.
So where the transcriptomics and the metabolomics and all those omics came up, I will not deal with those omics, but the question that came that individually when we studied transcriptomics, we would get a picture about the transcription rates, transcription processes within the cell, but it does not solve the problem whether there is any relationship of translation which needs to be taken up, or whether there are any post-translational modifications which could be need to taken up. An important thing which came up in between was whether metabolites do influence, whether they influence the expression of genes or even the post-translational expression of genes. And in fact, yesterday only we were talking with one of my friends about the role of metabolites on the expression of the genes, different genes and enzymes in different systems. And then whether there is any interaction between different proteins which express. So in short, it came to be called as the functional genomics, wherein it's not going. Okay. So different people started working on different lines, but I will try to take up something which I will not go into these details. I will not go into these details because these are already known. What we do with genomics, we have a sequencing approach. We finish the sequence, we annotate the sequence, and then after annotation, we then try to work out interactions. So what's important is what is the final goal of all these approaches? The final goal is not, as people try to think of, getting a sequence and trying to end up with a sequence. I don't know, Bionovid people would bear with me out that when we do even the transcriptome itself does not end up or give us much information unless we annotate the genes and then we do the linkages of different genes. So we will try to go out.
Kindly keep, sorry to interrupt, sir. I request everyone to keep their mics on mute.
Sir, I think you are on mute actually.
Again, he is muted. So Dr. Srungo, unmute yourself. You can do from your.
Okay now? Do you hear me now?
Yes, yes.
Yes, sir. Yes, sir.
Okay, okay.
Again, slide mode. Yeah.
Okay. So I will talk about something which is a bulbous crop, where we are trying to study how there is a transition from a state of vegetative growth to a state of reproductive growth. That is where we will try to look on the topic how the genomics has helped us. And also, this crop has flowers where it contains in the stigmas, it contains a specialized metabolite which belong to the group of apocarotenoids, protein and procytid. And we will try to understand how the pathways work out. This is just a picture to show the integration of the metabolic profiles between the dormant phase where the metabolic profiles metabolism is low, and during the events from transition from a dormancy to a vegetative start in renewed growth where the metabolic profiles activity start increasing, which then provides the substrate for carotenoid metabolism and the shikimate pathway, which then gives us the metabolites for flavonoid metabolism, oxygen metabolism, and all other metabolisms. So, is there a link between these? We will try to work out and try to see. So, what we approached was we had different comps at different stages of development. And as usual, you would do the transcriptome of all these at different stages. And then we all know we have to compare the data, we have to check up the validity of the data, we have to check up how much is the depth of the transcriptome and all those things, which we usually everyone does. If we do only that, it doesn't give us much of a picture unless we correlate the data with the phenomics. So in the case with reference to webinar that we have, the phenomics approach could be to compare it with the host and the pathogenicity, or how much the infection is there. In the relevant current talk in which I am speaking, our approach was to see whether our data is correlating with the differential phases of apical bud development. And as we can see, this is a dormant phase, and this is one of the most active phases where primordia have started developing. And then we have the other floral primordia coming up. So we tried to do this analysis, took out the sections, and noted the differential flowering development. We have the dome-shaped structure over here, and we have the floral primordia starting to appear over here in August. So between July and August, what is happening, especially in the apical buds? That was our purpose. And is that leading to flower development? So first thing that we did was to check up the growth hormone profile, and our data told us that at the dormant stage, we have a lot of abscisic acid. This is histochemical localization of immunochemical localization of abscisic acid. And as the floral bud development progresses, the intensity of the fluorescence decreases, indicating thereby a drop in the abscisic acid content, which is a growth inhibitor in the apical buds. So our question was, is abscisic acid individually responsible, or are there other factors also involved? We applied a promoter, growth promoter, that is gibberellic acid, and a growth suppressor, that was naphthalinistic acid, and we could see the differences in the apical buds. This is the control, and we see down there between 26 July of the control and the 26th July of the gibberellic acid treated, there is a marked difference in the primordial development. And we see the same thing on the same day of the naphthalinistic acid treated, there is a difference in the primordial component. There is a suppression of growth in naphthalic acid treated buds, promotion of growth in the GA3 treated buds as compared to what we find in the controls. So we did a metabolite profiling, and when we do a metabolite profiling, we find that in the different floral organs of the plant, we have differential accumulation of different metabolites. And primarily in the apical buds, it is the sucrose that gets accumulated a lot extent. If anybody would do it, they will normally do RT-PCR of specified genes and try to see whether that is working or not, whether there's a correlation. This routinely everybody does. The problem with this is that we don't get an integrated picture. So what we did was we did a transcriptome profiling of all the 3 cases at all the different developmental stages, combined the data, put up into a control form, and anyone would do the assembly report. And then assessing and after annotation, doing the fold change values, we get a picture. This is a typical picture of any transcriptome profile. Even now, if people do enzyme assays, they prepare a heat map. And that heat map gives us a picture of what's happening to different transcripts. There are 2 things which we have to take into consideration. Number 1, are all the genes relevant for further studies, or we have to now choose? And we get a lot of information for each of the different metabolic pathways, and these pathways tell us what is happening by and large within the picture. So the heat maps for different groups of metabolic pathways are prepared. We do prepare them, and we got a picture out of this, which gave us a situation of what is happening to each of the different enzymes. Like this is B1, B2, B3. This is the stage when it is quiescent. B1 is the quiescent stage, B2 is the active stage of growth has started, and B3 is now actively primordial development. We did a similar situation for the stigmas, where we have yellow color stigmas, orange color stigmas, and red color stigmas. Now we can see the difference in the picture. We have a highest sucrose phosphate synthase activity in the buds at the quiescent stage, and we have the highest sucrose phosphate synthase in the stigmas when they are red. So we correlated these with different profiles. This B1, B2, B3 was correlated with the developmental profile, and YOR of the stigmas was correlated with the specialized metabolite synthesis profiles. And we got a picture of the sucrose metabolism, starch metabolism, how the changes in these are related to the development. We got a picture of the glycolytic pathway, and this is all based on transcriptome profiles which were analyzed, and we correlated that with the stages of development. We got a picture about the TCA cycle. We got a picture about the, we got a picture about the glucose metabolism, glycolytic pathway. So we had everything with us about the carbohydrate metabolism. And why we took carbohydrate metabolism? Because the carbohydrates were expected to provide, carbohydrate metabolism was expected to provide the nutrients for the bud growth. We also now looked at the floral genes, and we have certain genes which are upregulated and certain genes which are downregulated in response to the treatments. So like APETALA2 is upregulated, more upregulated in the earlier stages. On the other hand, in the case of naphthalinistic acid treatment, it is not showing any change. On the other hand, LIV1 is down-regulated in naphthalene acid treatment, does not show any change in the gibberellic acid treatment. So we get a picture of like MIP. It is upregulated, but in the case of NA treated, it is showing baseline expression. MADS-Box is down-regulated in the case of NA treated. So we get a picture of what is happening within the tissues at different stages. And then based on that picture, we can follow an integrated profile of what must be happening when we correlate the expression profiles with different events or stimuli that might induce or trigger the transition of the vegetative buds to the reproductive buds. So some of the signaling which we correlated and which was correlating with our transcript profiles was involvement of hormone signaling, involvement of vernalization pathways, autonomous pathways. There were changes in the transcription factors, and there were epigenetic regulators also which were affected. I'll not go into the details for this for the paucity of time. So we looked at some of the genes, and our focus was on flowering locus. We had MADS-Box genes, and we correlated those with floral development. This was one aspect which we took up. This was based on the distribution of transcripts across different stages. There are transcripts which anybody will say there will be common to all the stages, common to all the treatments. There would be transcripts which will be specific to different treatments or different stages. Based on that, as we had shown earlier, we prepared the detailed heat maps. We identified the transcripts. We identified the changes. We identify which transcripts are showing differential upregulation or significant downregulation at different stages of development. So I will take, because this is a lot of data, which if I speak about, it will take a lot of time. But what we want to show is that it gives us a much detailed picture. And then we can compile the picture into one group of data sets as to which one's transcripts are showing differential change, what is the number, and whether they are getting upregulated in response to which stage and which change. The heat maps are all there. I will not go into those heat maps over here because I don't want to talk much about that. But all these give us a differential patterning. And we can focus on what is happening, and our focus again shifted to the AP2 transcription factor, which we now know that there are 3 flowering locus K, FCA, and constants like 9, which were shown to be, which appeared to be, I will not say shown to, which appeared to be directly involved in transition of the vegetative apex to the floral apex. So we did prepare a hypothetical diagram of what is happening, and this is all how the flowering locus FLC is causing the activation of processes leading to the transition of the floral apex, vegetative apex into the floral apex. Now, this was one part of the story. The second part of the story was that we had the transcriptome data and we correlated it with the transition in the stigmas from the yellow stage to the red stage, scarlet red stage. Now I will try to explain what I meant, and this is with regard to specialized metabolites. The stigmas, when they develop, initially they are yellowish in color. And as they mature and grow, which I showed in the earlier stages, they are scarlet in color. And the change in the color indicates a differential accumulation of certain specified metabolites which are accumulating only in the stigma. And these are crocin, crocetine, then safranal, and picrocrocin, which have been known to be universally important, which have universally been known to be important in several preventives for disease, like they have antihypertension, cardiac protection, they give relief to several pathogenic disorders in human beings. So, and also these components are essential as a spice, as a coloring agent, as a flavoring agent. So, what we noticed was that in these petals, we do get accumulation of anthocyanins, but in the case of stigmas, we don't get accumulation of anthocyanins. We get, on the other hand, much accumulation of apocarotenoids and flavonols, especially camphorol. Now, why this differential expression we wanted to study, and based on the transcriptome data, we developed linkage maps. This is basically networking of different pathways. And based on those networking pathways, we knew which metabolic pathways are providing the metabolites for the shikimic acid pathway, for the PAL pathway, and to what extent, as well as which are the metabolites which are likely to accumulate at what stage in the stigmas. So this was based on a heat map which we all prepare, and this gives us a picture of what is happening. The expression profiles have to be in any case validated by RT-PCR, qPCR, which we do routinely in these cases. If we stop at this stage, then it gives us a picture of most likelihood what is happening. But on the other hand, if we now use this data to move towards proteomics, then we can link the information generated from the proteomics approach and the information generated from the transcriptomic approach to give us a more clear picture of what is happening. And what we noticed that there are 3 transcription factors: MYC2, R3, R2, MYC12, and TTG1, which show significantly differential expression in the stigmas between the orange color stigmas and the red color stigmas, also between the quiescent or dormant apical buds and the stigmas. We did a structural analysis of these and noticed that MYC2, R2, R3, MYC12, and TTG1 do interact with each other to form an MBW complex. Now, this MBW complex acts as a regulator for the activation of genes responsible for the synthesis of anthocyanins. Now, we also noticed the difference between the apical buds, which were quiescent, and the stigmas, that in the case of apical buds, we had a much lower interaction happening, and well as because of the surface area issues, as well as we had an expression of another transcription factor, R3-MYB, which is expressing itself more in the stigmas at the red stage than in the stigmas at the orange, yellow and the orange stage. When the whole thing was analyzed, the interaction studies provided a picture, what's happening, that when we get a pathway, that is what I want to tell here, that our data tells us that we have a pathway which starts with PAL, coumaryl, coenzyme A, naringin chalcol, dihydrocamphorol, and the diversion. The diversion is because of a DFR as well as FLS. So the relative activities of DFR and FLS, I will say the ratio of the activities, that will determine whether the metabolic pathway will be processed towards or will be diverted towards anthocyanin biosynthesis or will be diverted towards the flavanol or the camphorol biosynthesis. Now the question is, what regulates this step? And that would give us a picture of what's likely to happen. What we noticed was that if we have a stable MBW complex, the stable MPW complex promotes the suppresses FLS activity and promotes DFR activity, which leads to the pathway diversion from dihydrocamphorol towards leukoanthocyanins, leukoanthocyanidins, and anthocyanidins, which are then transported into the vacuole to get accumulated as anthocyanins. And this happens, we can see the transcript profiles active. This happens when the complex is stable. On the other hand, if we have in the picture this ETC1, if this expression is increased, which increases in the case of red stigmas, and this is the situation which happens in the case of orange stigmas. But in the case of the red stigmas, when we have a ETC1 that is 1 and MIC1 that is another inhibitor, when we have this, they do not allow the formation of the stable MBW complex. Now, these have been reported in the literature as MBW complex interferers, and they regulate the stability of the and the formation of the MBW complex within the cells. And when the complex is unstable, it promotes the synthesis activity of expression of FLS and the activity of FLS subsequently, which then diverts the dihydroxycamphorol into the camphorol biosynthetic pathway, and ultimately we get the flavanol rather than the anthocyanins. Now this gave us a picture of, even though we if we study the transcript profiles independently, it will give us a picture but will not be able to tell us about the regulatory factors. And if we study each of these regulatory transcription factors independently, their expression may, in certain cases or in certain other cases, may not match with the expression of different genes because of the interactions that are happening amongst these proteins. And if the interaction is stable, it may regulate the expression of a different gene, and if it is unstable, individual transcription factors may regulate the expression of different genes, which is important. That's happening for the synthesis of the specialized metabolite, which we are talking about in this case. Now, this was in brief what I talked about, and this is the team. Amongst this, we have this girl is right there itself. We have Upasana, who worked on this problem. We have Sudita, who worked on this problem. We have Jyoti, who worked on this problem. And we have Mona, who worked on this problem together to contribute. This is just a brief of what we have done to contribute towards understanding of the flavonoid biosynthesis pathway in saffron stigmas, as well as the developmental pathway leading to the transition of vegetative apex to the floral apex in this system. I hope I finished my talk in time so that if there are any questions, I would be most willing to respond. Thank you very much.
Thank you so much, sir. The talk beautifully summarized.
Can you be a little bit louder? I am not able to hear.
Yeah, thank you so much, sir. Your talk beautifully summarized the. Hello, am I audible? Am I audible?
Yeah, you are audible.
Okay, so thank you so much, sir. Your talk beautifully summarized the various omics-based applications in deciphering all the intricacies of the various metabolic pathways. Now I would ask the participants to kindly come up with questions in the chat section so that we can put them across to our speakers. kindly drop in your queries in the chat section. Um, Mr. Deepak Sharma is appreciating the talk, sir. He's saying it was quite an informative talk. Um, Dr. Parvez Ahmed Sheikh is also saying it was an excellent presentation. He was thanking you for the same. Any questions apart from all the compliments? Any more questions, guys? Please, you can please drop them in the chat section. Sir, we have a question from Dr. A. Ratan Kumar Singh. He's asking, is there any potential crop to exploit metabolites from northeast regions?
Okay, very good question. I did not share, for paucity of time, another aspect of the study which deals specifically, which deals specifically with the Northeast and more specifically with Manipur. In Manipur, we have black rice. And black rice is known for several of the positive health benefits, especially the amount of different specialized metabolites. We got some accessions of black rice from ICR itself. And what we wanted to do was, why is it that rice as such does not accumulate carotenoids? Because that has been going on for quite some time. We are all aware about golden rice and others, which are transgenic approach, which was used to up the accumulation of these carotenoids, vitamins in the rice cultivars. So we have a detailed study on that, and we have been able to get, I will say, 2 accessions which accumulate more of beta carotene than even the golden rice. And we did work out the reasoning using the transcriptomics and proteomics approach to identify the rationale as to what is happening. I did not talk about that, but definitely there is a lot of, I will say, germ plasma available in this region, many plants available in this region, which have medicinal values, so they have specialized metabolites. And the omics approach, I will call, together can be used to decipher the metabolic pathways and then use that information for manipulating the pathway to increase the biosynthesis of or accumulation of those metabolites in those very plants rather than going for a transgenic approach. So there is an excellent possibility, and people do need to work on those aspects.
I hope it answers your query, Dr. Ratnakar. Ratan Kumar, sorry. We have a question from Dr. Binod Bihari Sahu. He's asking, is there any metabolite active compounds from plant to control plant pathogens for field application?
Oh, now then we need to, okay, I will give you that aspect. We need to then isolate these metabolites and test them, but in vivo, these metabolites, many of these metabolites are known to act in defense of the plants. Say, I will give you one example. There's a crop growing in this region. We have worked on some buckwheat. This is grown in the Himalayan region, and it accumulates a lot of quercetin. And quercetin is an antifungal agent. It confers the resistance to the crop towards pathogen infection. These are possibilities, but basically the issue is to take up the information from the basic research, apply it using the molecular tools, and then translate. So translation is important, which unfortunately, at many places is not given that much importance. But yes, there are.
Thank you so much for the answer, sir. I hope it satisfies your query, Dr. Sahu. We have another question from Dr. Deepak Sharma. He's saying, I have read that only corns more than 8 gram produces flour. Is there any specific reasons?
Yeah, there is a relationship. The smaller the comb size, the smaller is the size of the apex, shoot apex. And this has been found to have a tremendous influence on the development, or I will call the initiation and development of the leaf and the floral primordia. The leaf primordia do develop, but somehow the floral primordia don't develop. We have noticed this. We have noticed this thing ourselves also. Smaller size combs don't produce flowers. So somehow the floral primordia don't develop. My own hinge, I have not done any work on that aspect. My own hinge is that the smaller combs are not able to provide the required amount of uh, assimilated products to the developing shoot apex, and that causes the abortion of the developing floral primordia in the initial stages itself, and that's why the floral development does not take place. This is my own hunch. I might be wrong on this, I do not know, but this is a fact. Smaller the size of the comb, the number of flowers decreases, and below 8 gram, I will say 8 gram, I will say okay, 8 gram, well, floral development is significantly obliterated.
Thank you so much for your response, sir. We have another question from Dr. Preeti Pathak. She's asking, how can we fill the gap between research and applications of these research to the field? The second part of her question is, what should we do if pathogens are resistant to biological or chemical control?
Well, find out other ways of controlling. There are many proteins which do show antifungal activity, and that I think is Sushil, in your lab also, the girl has worked on that aspect. We can try different, not only the biological activity, we can try different proteins, and there are antifungal proteins which are available. Uh, Go translate them in vitro and try to produce them in bulk and see if these proteins do have the same level of antifungal activity against specific pathogens, because these cannot be broad spectrum uh, in totality. So use them. That could be one. Or the other way is to use transgenic approach to develop resistance against the pathogens, and that is also used. Develop new varieties. So there are, but the problem is that it have the basic information and translate that. How to translate that into active, productive use? That's the question which needs to be answered.
Thank you so much for the response, sir. Uh, any more questions? The participants, please drop them in the chat section. We
So
still
Avinash,
have time in hand. Yes, sir.
Avinash. So like what Professor Churungu has been answering, as he has said, along with his lab, we had been working on identification, isolation, purification of beta glucosidase from cesium, which is called chocho, as sir has said. So we are working on its antifungal and antibacterial activity. So in silico and in vitro studies has indicated its potential use as antifungal activity, but then we are purifying it in the pure form, and we are trying that how its formulation can be really made so that we can use it for application in the field for controlling the diseases caused by fungi and bacteria. Thank you, Professor Churungu, sir.
Yeah,
Thank you very much.
And
thanks for the insight, sir.
yes,
I think we are done with the questions. I don't think there are any more questions coming up.
so we must thank Professor Churungu for accepting our invitation from the behalf of Indian Phytopathological Society and BioNIBI and such a elaborative and informative talk. Thank you, sir, for sharing your valuable time and be with us this morning.
Thank you, Sushil, and
Thank you,
thanks
sir.
everyone
Thank,
for giving me the time to be here with you and interact with you people.
thank you,
Thank
sir.
you. Thank you.
Undoubtedly, sir, it was really an informative session. Thank you so much for your time, sir. Thank you so much. Uh, Dr. Sushil, we have a small question for you in the chat. It's from Dr. Amrita Banerjee. She's asking, may I ask Dr. Sushil against which fungi you are testing?
Madam, actually, we have only tested it in vitro against your Magnapore theorizi, Rhizoctonia, and Fusarium.
Okay, I hope it answers your query, Dr. Amrita.
Yeah, thank you, Dr. Sushil.
So, sir, shall we start with the next talk?
Yes, yes, you please go ahead with the next speaker.
Okay. So after this insightful talk from Dr. Trungu, let us start with the next session. So non-host resistance. Okay, we have a Dr. Sushil. Just a moment. Sir, we have a question from YouTube for Dr. Trungu. Is he online?
Sir, I think he's online.
Yeah. So this is from.
Sir is there. Yes, he's
This
there.
is this is asked by Dr. Ms. Deepu Pandita, and she's asking, have you worked on Kashmiri saffron only or also Kishtwari saffron? Uh, this is her question.
Okay, nice question. There is no difference in the aspects which we are talking about, which we are working between saffron from Kishtwar and saffron from Kashmir. The metabolite profiles do not show significant changes or differences. There was used to be a time when it was considered that due to certain reasons which were unknown at that stage, that saffron will grow only at a particular place, and that is Kashmir. But then saffron cultivation has been extended to several regions, including Himachal, Kashtwar, and I must say here, in experimental conditions, we grew it in Shillong also. So whether you talk about Kashmir saffron or whether you talk about Kishtwar saffron in terms of developmental profiles, because that is what mattered for us, there is no difference. We have seen the samples from both the places. Does
Thank
that
you.
does
I think so. Yes. Yes. It answers her query. Thank you so much for this for answering this last minute query, sir.
it's okay. It's good. It's good if there are questions. I like it.
Just let me confirm once whether there are any more questions from YouTube or not. Yeah, so I think we are mostly done with the questions. Thank you, sir. Thank you for this last minute answer. Uh, so I think we should start with the next talk. Non-host resistance is a broad spectrum plant defense that provides immunity to all members of a plant species against all isolates of a microorganism that is pathogenic to other plant species. Our next speaker, Dr. Binod Bihari Sahu, who is an assistant professor at Department of Life Sciences, NIT Rourkela. With more than 25 publications and a patent to his credit, Dr. Sahu's research interests include plant immunity, non-host resistance, during a plant pathogen interaction and the mechanism of plant cell death. Dr. Sahu would be sharing his views on identification of non-host resistant gene from Arabidopsis and its deployment for soybean. So I would like to take this opportunity to invite Dr. Sahu to share his views on the said topic. Over to you, Dr. Sahu.
So may I request Professor Churungu to kindly unshare the slides so that Sahu sir can share his slides. Avinash, am I audible? Yeah. Yeah.
Yes, sir. Yes, sir.
Yeah. So now you may request Dr.
Yeah.
Sahu to share his slides.
Dr. Sahu, I would request you to kindly start screen sharing.
Thank you. My screen is visible.
It is not as of now. Yeah, it is, sir.
Yeah.
Yes.
Thank you for inviting me for sharing some of my research work in here. So I thank Indian Phytopathological Society and BioNVID so providing me the opportunity to present the work over the common forum. So my topic for today's discussion is identification of non-host resistant gene from Arabidopsis and its deployment for soybean immunity. Before I go into that, let's me be little bit discuss on the plant pathogen interaction and how the defense system works. Although it must have been explained in the previous lectures. So basically, when the pathogen actually, so as you can see in this slide, so many types of the pathogens are there, starting from the fungal pathogen or the bacterial pathogen or some insect or the those one which actually kill the particular plant for their food material. Okay. In turn, what happens? The plant try to defend itself in the format of they can provide antimicrobial compound or some phytohormone influence can be happening, and also by plant-to-plant communication. So that priming might be also working. Okay. In that way also plant also try to defend itself. Okay. So at the same time, so intra-plant communication also can happen, but against the although insects, indirect defense might also come up. Okay, and so also some other pathogen can come to counteract the different kind of the different pathogen basically. Okay, so basically plants do not just surrender to the infection by the different pathogen, also the insects. Okay, so basically if we go to check, so how the resistance is really provided is that like mostly which is mostly on the basal and the non-host resistance. Okay, so basal is just the ability of the plant which can reduce the severity of the disease which are caused by the adapted pathogen. It means like let's say one particular your plant is affected by a group of the pathogen, like let's say soybean is infected by the Phytophthora sojai, and so also like rice is infected by the blast disease which is caused by the Magnaphorthia rhizae. So in that way the plant will be trying to reduce the severity, okay, so with its utmost potential whatever it has in the innermost part. Okay, but the pathogen knows how to subvert the whatever the basal resistance is there. Okay, downstream what happens, the pathogen again secrete another set of the molecule which I'll be discussing in the common next slide, so which will be really explaining in much more detail. Okay, so the other platform of the your resistance is non-host resistance. Also the resistance gene based resistance are there, okay, which are actually not that durable. That also I'll be discussing. Okay, first of all the passive defense is there where the pathogen attacks, okay, and where the plant side preformed inhibitors like antimicrobial proteins or antimicrobial compounds are there which are actually inhibits pathogen. Okay, but if we see the active defense or the inducible defense upon the perception of the pathogen, what happens? The particular compounds they enter the plant, okay, and then so they are received by the receptors on the plasma membrane, and downstream those signals will be activating the defense response, okay, and those will be activated like activating the R genes mostly, okay, and in turn that will be actually activating the resistance response, okay. So this is a myriad of the so much of responses as I told in the previous slide concerned, okay. So the plants, they do have the immune response and which act as just the single cell layer. It's not that so much of humoral cell lines like the innate immunity which is really working in the animal system that really works, but in the plant that what happens, just a single cell where the infection or the entry point, so that cell itself does the job for the immunity, okay. And for that, like let's say you can see in the slide, so there are many type of the receptors sitting on the plasma membrane, and which are the receiving end, okay. Like let's say in the from the right hand side, if I can discuss, so many type of the signals which comes from the pathogen are received by the your pathogen your PRR molecules are right. So or the PAMP recognition receptors are there, okay. So upon that receiving the signal, so downstream signaling cascade like followed by the MAP kinase assay, MAP kinase those cascade, the signal goes to the nucleus, and which really reprogram the uh, uh, transcriptome to activate certain type of the R genes, right? And those R genes, so in addition, so also activate chloroplast, peroxisome, and other mitochondria, all those proteins, and in turn, so that will be activating the defense response. Okay, defense response in the sense. So finally, in turn, that will be actually killing the pathogen at the entry point. Okay, whatever the means, it may be so by the means of the H2O2 or the callose deposition or some other molecular expression or some killing of the pathogen by any means, okay. So that really happens. So okay, so but in here what my research is actually concerned is so mostly non-host resistance, okay. If we so describe the particular terminology, how it will be explaining is it's mostly the phenomenon, okay, of the entire plant species which is resistant to all genetic variants of the non-adapted pathogen species in turn, okay, and it has been accepted by the common forum. And in the plant system, actually it is the that defense system is in a zigzag model which can be really explained. First of all, what happens, the pathogens, so they secrete the PAMP molecule or the pathogen associated molecular patterns. Earlier it was called like DAMs also, and upon that one, what happens, the pathogen, so with its preformed barrier, it tries to defend itself, and thereby it goes for plant triggered immunity, okay, followed by if that immunity is really there, okay, from the plant side, then the pathogen try to secrete this second set of the effector molecules or which are virulent molecules, okay, which the plant might be so not really realizing that okay some infection is really happening, but in that time what will be happening that effector trigger susceptibility will be coming up, okay. So that means the plant will be so surrender to the pathogen, but the plant actually does not surrender in just like that quite quick moment, okay. For that the plant actually try to express the R proteins against those particular effector molecules, okay. So that will be really recognizing those R molecules, okay, no, those virulent molecules, and the R molecule will be going for a cognate protein-protein interaction, thereby activating the hypersensitive response, okay, hypersensitive response in the format like let's say H2O2 or the active antimicrobials will be really expressed, and then the pathogen will be killed by, okay. But further, the pathogen still does not say that okay I just surrender to that, okay, but it express the second set of the your effector molecules, okay, and for that again the susceptibility will be happening, and further so towards the new type of the effector molecule, the plant will be trying to express the new set of the resistance protein, and this warfare or the plant pathogen warfare keeps on going until there is a conclusive end that okay so on someone is surrendering or not, okay, and that also takes so from time to time, so like let's say 2 days, 3 days, up to 6 days, or it also keeps on going, okay, unless the warfare is going to be a stop, okay. So as I explained the non-host resistance, okay, so here So for a common example, like if we can say that soybean is a host to Phytophthora sojai, whereas Arabidopsis is a non-host. Means Arabidopsis is not actually infected by this particular pathogen which really infect the soybean. Okay. So also like rice blast is causing disease to rice only, right? But it really cannot cause to the plant like Arabidopsis, like let's say mango plant, it's not going to infect. Okay. So those other plants which are not infected by the particular pathogen are actually told to be non-host. So something extra in those non-hosts are there which actually play a vital role for defending themselves, right, against those pathogen intrusion. So far what has been already identified are at a different layer of the defense, right? So this resistance mechanism really works. So at both pre-hostorial as well as post-hostorial level. Okay, pre-hostorial there are 3 genes which have been really identified, like PEN1, PEN2, and the PEN3. As you can see on the slide, upon the pathogen, when the spore really landed, okay, then the apressuria is really formed, and then it try to penetrate this cell, right? So at that moment, what happens? The PEN1, right, or the syntaxin, so are the one which really release those particular or the your callose deposition, right? And further downstream, the PEN2, the active redox or the material which really are against those pathogen will be traveling to the site of the infection via the PEN3 molecule, which are the binding cassette or the transporter, which lead to depositing those antimicrobial compounds to the pathogen, so that it is really going to be killed. Okay, further downstream, those recognition signal is going to really activate this particular PEN1, PEN2, and the PEN3 molecule, so which act as the pre-hostorial level. But what happens? Suppose this particular one is really defended and your pathogen really enter the cell, right? And it makes a hostoria kind of structure, okay? And then it invaginate inside the cell, okay? And then the effector molecule from the pathogen are really secreted inside that of the cell of the host, okay? And then, which are all signaled into the nucleus, and downstream the activation of the other NHR genes, which are the non-hostile like PAR4, EDS1, SS, or the SAG101, which codes like for PAR4 codes for lipase like gene, EDS codes for again the lipase like 5th gene, okay, and SAG is coding for a acyl hydrolase. Okay, similarly, so this work like in the plant itself, there are many layers of the non-host resistance itself. Okay, and so far, so many of the people are reporting the group of the non-host resistance against different kind of the pathogen. Okay, so and we actually studied the stuff in the soybean disease, which is caused by the Phytophthora sojai, and it's a $1 million loss to the farmers. Okay, and as we already also know that for the plant pathogen interaction, the plant try to express the R genes, right? So for the against the P sojai, there are many RPS genes which are the R genes against this particular pathogen. But the problem is, although they are effective against those pathogen, but that one is effective only against a set of the race, but it is not durable. And over time, what happens if this resistance gene is already deployed in the field? What happens? The pathogen try to mutate itself, right? And thereby the new version of the equivalent or the effector molecule are secreted, and then the crop is really affected by this pathogen, right? So that's why we are not saying, we are saying that it's not durable. Okay, for this one, then we wanted to test whether this non-host resistance gene can be identified and can be deployed into the soybean to make it resistance, and which will be actually like broad spectrum and will be providing durable resistance, right? So for this, what actually, our work plan is, we had to generate the mutation in the Arabidopsis, which is already mutant, which I will be explaining in the further slide. So this one I'm just going to give an overview of what I will be telling in the further slides. So we'll be having the mutant, then which will be crossed to another wild type resistant variety, that is nidogens, and the F1, which is going to be a heterozygous, and is going to be also a resistant one, will be going to the next generation, that is F2 generation, and then there will be screening for this susceptibility phenotype. So and there, so as per the ratio, whatever the phenotype ratio, the resistance or the susceptibility ratio, from that we'll be going for so map based cloning and the NGS for the identification of this gene. Okay, and further we'll be validating that one. Okay, and further upon integration into the crop plant, that is soybean in our case, so we'll be checking whether the resistance gene is really providing resistance. Okay, and then so if we venture into our research, what actually we did is that we can see there are 4 pictures on the top bottom. Okay, so top portion, so that is showing Colombia, NHO1, PEN11, then PSS1, right? So in the Colombia, so as you can see at the entry site where the pathogen actually landed, so that one is showing so the appositions or the callose deposition where the particular defense is actually there. Okay, but if we go to the next one, right, NHO1, that is a mutant in the NHO gene, where the similar kind of the resistance response is clearly visible. But in the PEN1, you can see there is a single cell death, right, where the pathogen could enter the single cell. But so if we go to the next one, right, PSS1, which is generated so by our group only by EMS treatment, so that further so mutated in the PEN1 background, and further we can see that the pathogen actually could see the multiple cells entry. You can see those trypan blue stained cells which are multiple. Okay, and the bottom side, so you can see the phenotypic analysis, how we can really see from the outside that how the leaf is really looking like. Okay, so the nitrogen is shown. Okay, so the macroscopic view, you can see the spore where it is landed with a drop, like we put 10 to the 5 spores per ml. So 10 microliter we have put, and you can see there is no such cell death. But in the PSS1 mutant, you can see the yellowing is happening. Okay, so also the cell death is really happening, the darkening of the cell or the blackening of the cells on the top layer is really clearly visible. So the microscopic picture is really clearly visible. So in here, so I think so there is some problem. Somebody wrote something, how to erase. On the screen there is something red. I don't know, maybe I can proceed. So anyway, so if I proceed ahead, then so actually what we did is we further characterized those mutant that we generated. It means in the previous slide that I told, we generated PSS1 mutant, right? So this PSS1 are so having multiple cell entry at the beginning, and then we cross-checked that one for the defense pattern. Okay, so we stained with the aniline blue, so which will be really detecting the callose deposition. Callose are the molecule which are really secreted at the site of the point of the entry by the pathogen. So in the Colombia, as you can see, there are very few cell entry points, so which is really so kind of showing the resistance response. But in the PEN1, there are very few cells which are really just by entered by the pathogen. Okay, so or the entry site, so which is really so responding to the pathogen. But in the PSS1, as you can see, there are multiple cells which are really entered by the pathogen. As you can clearly see, so many of the callose deposition sites are really visible. But the bottom one, B picture, so is showing the trypan blue so staining or the trypan blue exclusion assay, where the trypan will be really taken, is not going to come out if that is going to be cell death. Okay, in the Colombia, there is very so are the just entry point, just the appositions are there, so that's why the pathogen some structure are only visible. But in the PEN1, you can see single cell so death are there, but in the PSS1, just within 3 days, the secondary life cycle is already over, right? The next cycle of the sporangia are formed from where your spores are already so generating. Okay, so further what we so actually wanted to do is that, so we wanted to say that, okay, the PSS1 upon mutation is really so now breaching the immunity to this PSOGI. Okay, for that what we did is we crossed this PSS1 to nitrogens, which is a resistant one. Then we got the F1, which again we tested by the phenotype, which I have not shown the result. Okay, so but we saw the resistance response, right? Because it is in the heterozygous condition, that gene is not really going to be expressing. Okay, so then so So we went to the F2 generation, and in the F2 3 recombinant inbred lines, we just checked for the phenotype by screening or by infecting in the detached leaf infection assay, as I saw in the previous slides. So here you can see in the table as like the observed and the expected values are shown. Okay, for homozygous resistant plant, we got 12 in number. Heterozygous one, SS1 and small ps1 or the little allele or the recessive allele. So that time heterozygous one we got 21, and homozygous susceptible we got 9 plants. Okay, out of the total 42, as per the calculation, the chi-square value is 0.43 and p value 0.81, and it's really clearly suggests that it is really following to a 3:1 Mendelian ratio, and which is going for the recessive mutation. It clearly says that, so this PSS1 mutation, whatever is happening, so and that phenotype which is associated is governed by a single recessive gene. Okay, but the question might be happening that as we have generated the PSS1 mutant under the PEN1 background, so might be PEN1 is also playing a role, but in order to avoid that, we just tested by marker analysis and checked that, okay, this PEN1 marker so is segregating along the randomly selected recombinant inbred lines. As you can see, the PEN1, so in the marker portion, PEN1 is showing a lower band, Columbia is showing a higher band, and the heterozygous it will be showing both band. Okay, but as you can see, the recombinant inbred lines, there are all sorts of the banding pattern, higher band, lower band, and the both band means, so A is going for the PEN1 type, B is showing the Columbia type, H is showing the heterozygous. So it's a kind of still it is kind of segregating. So we conclude that this PEN1 is really not functioning in the background. So with the PSS1, or it is, we say genetically that PEN1 is not epistatic to PSS1 gene. Okay, so then we are now sure that, okay, PSS1 is the only gene which really provides so resistance against the physozyte. In order to really find out what is this gene exactly, right? So can we really map it and identify that one? So for that, what we did is, so before going to that one, we really checked whether this resistance is broad spectrum or only specific to this pathogen. Okay, we further tested those susceptible and the resistant plants which we identified against the physozyte, tested against the fusarium, which causes the sudden death syndrome. That is a necrotrophic pathogen. Okay, but the physozyte is a hemibiotrophic pathogen. So as you can see in this slide, see picture. So Fusarium virguliforme. So that case, the resistant plant are also showing resistance, and the susceptible one which are breached so with the mutation are also showing the susceptibility response to the Fusarium. But whereas if you see the B picture, which is really showing the infection strategy, or the picture from the P. syringae infection, right? So this one is kind of segregating all sorts of the pattern we can see. Okay, the susceptible resistant or the resistance plants, or the susceptibility means that is written by S or the R. Okay, or the R is showing the resistance one. Okay, so this particular phenotype is kind of segregating. So we say that the PSS1 is really required for non-host resistance in arabrosis against the P. syringae as well as the Fusarium virguliforme, and these are the two fungal pathogens, but it is not providing resistance against the Pseudomonas syringae, which is a bacterial pathogen. Okay, so then further, so we wanted to actually map that gene so onto the arabrosis chromosome. Okay, so for that, when we actually checked, so that against Columbia and Niedergren's cross, okay, there are not many markers which are evenly placed onto the chromosome. Okay, for that, what we exactly did is we went for the whole genome sequencing of the Niedergren's plant. Okay, which actually is a providing immunity against the physozyte. Okay, and upon getting the next generation sequencing data, we went to align that data onto the Columbia sequence, right, or the reference genome at the TIR10 sequence, or that particular release or the recent release, and then we identified the SNPs which are governed onto the different or the across the genome. Okay, and as per the SNP, we defined or we generated SBP marker or polymorphic molecular marker which can really distinguish the different regions onto the chromosome. Okay, and those places onto the chromosome where there are no such marker which can really delineate or the difference different those two accession. So we designed those SBP marker, as you can see on the chromosome map. Okay, 18 codominant SBP marker we were designed. So based on the NGS data, which was really done by us, and it was also published, as you can see onto the chromosome 1. So the wherever SBP is writing, right, so that is showing a SBP marker. Okay, also the other chromosome we developed so many markers. Okay, and we published that one. So then further we actually use those molecular markers to find out the place of this particular PSS1 mutation wherever it is lying. So by using bulk segregant analysis. What is that? So we actually bulk the genomic DNA so from these susceptible plants that we got from the screening. Okay, so and we made 4 bulks in here. As you can see, S1, S2, S3 and S4 are the bulk which are derived from the susceptible plants from different combination. And then you can see also Columbia, nitrogens and resistant bulks are also shown in the picture. Okay, and as we can see, so onto the chromosome 3 lower arm, so this particular gene is really mapping. As you can see, NGA707 is looking like a Columbia. Okay, or it is the background is like Columbia because the PSS1 mutation is really made in the Columbia background, right? But nitrogen is the other counterpart or the other resistant plant which actually was crossed through. Okay, so the susceptible plants wherever is there in the F2 generation should be susceptible because that point it will be really homozygous for. Okay, so if that is really localized to NGA707 is really looking like the Columbia type of the binding pattern, but other markers are showing heterozygous, so or the other type of the nitrogens type of the binding pattern. So we say that this particular gene is really linked, tightly linked to the NGA707. And further, so we also did the fine mapping using the other sort of the SBP marker designed in this particular NGA707 in north and south region. And then we actually found that onto the chromosome 3. So within 2.69 megabasphere, this particular gene is really localized. Okay, and further, so to actually find that which particular gene is there is 2.69 megabasphere means there will be really like so many genes in this place. But we from the screening assay or the your Mendelian that ratio, we got that it is only single gene. So in this 2.69 MB, there will be many genes. Okay, for that to identify which exact gene is there, so we went for next generation sequencing. Okay, and there we actually got 6 non-synonymous SNPs in 6 genes which were identified from the next generation sequencing. Okay, which actually was clearly seen from the IGV platform, which actually we could really see. Okay, and for that actually we run so many programs from the next generation sequencing data. First of all, first QC was really called for using this particular command. As long as you just know the simple Linux command, so you are good to go, and you can do at home also, no problem. And then so we use the simple pipeline, just three command lines, right? So this is the command line, so cd simple chmod, and this create for the simple .sh, so which will be really giving to align to the reference genome, that is Columbia, okay, or the TIR10 release. And then further the IGV that I shown in the previous slide, like in here you can see, so bottom side, so IGV is the platform where we can see where the exact SNPs or the mutation, whatever is there, okay, right, upon the alignment. Okay, and then BAM to BCF conversion was also done using this particular command, and the variant format was generated using the which was converted to NGM, so which is actually showing like whether one particular region is heterozygous or homozygous, or like both parental combination are really shown. So those commands is really going to show. Okay, online platform you can clearly visible. Okay, so basically what we identified is, so the next generation sequencing and the analysis by the simple pipeline revealed 6 candidate genes in the mapped region of the PSS1. Okay, what are those genes? So those are tabulated in this particular table, 6 genes, glycine rich protein, and the gene ID is just written in here. Also, so for this actually we did this SOR mapping for this to really confirm whatever we got this pipeline is all correct. Okay, so this glycine rich protein, which is found also in the exons, okay, and the mitochondria ribosome protein, unknown protein, and the microtubule associated protein contains the interprodomain, fatty acids, sphingolipid, desaturated genes. So all the genes, so this SNP position is found in the exon. Okay, So, but before going to really see that, okay, this gene is really present in the soybean or not, so we wanted to check the orthologous genes in the soybean. And as you can see by the blast analysis in the identity region, so slide, you can see not more than 40% of the similarity is really seen in the soybean. Okay, so it means, so clearly this arabidopsis genes, whatever we found, these 6 genes, let's say 6 genes we are getting, so might be, so more or less they are quite different than that of the soybean. So we say that, okay, this particular gene, whatever we found out, so might be really helpful in providing the resistance. But so as per the ratio and stuff, still we are in confusion whether it is a single gene or not. Okay, but it must be a single gene. Then, so out of the 6 gene, which is the gene that is really providing resistance? For this, what we actually did is, so we ordered the mutant lines from the Arabidopsis. So already those mutants are available in the public domain. Okay, so we ordered those mutants against these particular 6 genes. Okay, and found out. So okay, so this map actually shows onto the chromosome 3 lower arm. So those particular 6 genes, whether they are going in the right orientation or the left orientation, those are shown. Okay, so and then so only for the top protein, which was in the table, that is glycine rich protein, was the only one. So mutant of that particular gene only showed the susceptible response. Okay, so actually we got the Salk lines, which were actually 2 were found on the promoter and 1 is in exon. Okay, and all kind of showed the susceptible response, but not the others. So we conclude that, okay, this is the gene which is actually providing the resistance. Okay, so for this actually we wanted to really check those tDNA lines, whether the expression is still there or not. Okay, as you can see in the D picture, so which is actually falling in the exons, so the expression is not actually there. Okay, that particular gene expression is not there. So also the C picture, where the one of the promoter GRP2 is there, so there is no faint expression is there. Okay, that's why the susceptible response was little bit. Okay, but in the promoter 1, the first one, GRP1, so some sort of the expression is still there. Okay, that clearly shows that, okay, maybe the resistance whatever we are getting is due to this particular gene. Okay, so this is the one to just check the tDNA insertion, whether it is homozygous or not, and it is homozygous mutants. And this is the particular gene, what are the intern, exon, and the promoters, all those are just shown. And we wanted to actually clone this one, clone this particular gene. So the genomic DNA as well as the cDNA, genomic DNA including the promoter. Okay, cDNA is only starting from the start codon to the stop codon. We just wanted to amplify and clone into the binary vectors. Okay, so this is the cDNA which is really cloned into. Okay, and the star places shows actually the confirmed clones which are having the gene insert. Okay, so this is the cDNA cloning into the binary vector, that is the PISU agron, where in the biomatron site was the one which we actually created this particular binary vector. And so also we checked the orientation, whether the orientation is really all perfect, so forward to reverse. So this one only clone number 9 was the one which is actually so right orientation. And further, so we checked the genomic DNA cloning also, so that also we performed the similar pattern, but that vector was PTF101, that is another so vector where the 2 times 35S promoter are there, which is really vivid, so which will be expressing the protein to a greater extent. And then further what we actually checked is this is the result. Okay, so as you can see that this particular Arabidopsis mutant or the PSS1 mutant, okay, upon complementation or upon interrogation of the wild type Arabidopsis gene, that is the glycine rich protein gene, so now it is showing the resistance response. So also this Salk line or the mutant that we checked for this particular gene only, upon complementation are showing the resistance response. Now we are really confirmed that, okay, this is the gene upon mutation is now showing the susceptibility response. Okay, so then so we wanted to really characterize that one. For that we actually wanted to say that, see by bioinformatics analysis, that by transmembrane domain it is actually falling onto a, so having some transmembrane region. Okay, so it means it is found onto the cell wall or the plasma membrane, that particular so domain. Okay, so and then the expression pattern was also checked. Okay, as per the different time point of the infection, 0 hour to 96 hour. okay, and 6 hour to 36 hour, there is huge expression. It is going more than one-fold than that of the background, okay. And then also different tissues were also tested, and in the silics or the your pods, okay, so the expression is really higher. And also we checked for the gene ontology enrichment, where the log 1 by p value was really calculated for the co-expressing genes, okay. The co-expressing gene means already the established database were searched for what are the genes that really co-express along with this particular gene, and we found out that by the classification or the ontology, so the autophagy genes are to the higher extent they are expressing. So also the protein which target to the vacuole and the vesicle mediated transport genes are really also activated, so along with this particular gene, okay. Then this particular gene is really localized to plasma membrane or not. As per the prediction, we also tested by GFP tagging and the M-cherry, which is a marker for the plasma membrane. And then upon the merging, you can see the PSS1 so is really falling onto the membrane, okay. And upon plasmolysis also, you can see in the bottom the B picture, you can see along with the plasma membrane only they are just coinciding, right. And so that clearly really showing that this PSS1 gene upon expression or in general in the plant itself is really localizing to the plasma membrane, okay. So then further, so we wanted to test, okay, although we found out this particular gene, whether this can really provide resistance in the soybean itself, because the soybean pathogen was really tested. So then for that, we actually used the promoter 1, promoter 2 and the ubiquitin promoter and cloned this particular gene. Promoter 1, promoter 2 are the two promoter which are actually inducible expressors. I have not shown in this one. So okay, and then so under the ubiquitin or the all the your continuous expression under that particular promoter, we cloned this particular gene, and the bar is the gene for the selection purpose. And then we checked in the plant growth chamber that this resistance response was also clearly visible, right. And you can see upon finding those transgenic soybean plants, right. So you can see those resistant plants are now clearly so providing so much of the resistance, okay. So the wild type is, as you can see, so are showing so much of susceptibility, whereas this generated transgenic plant under different promoter are showing the resistance response, okay. And you can see so this PSS1 gene, the expression in the transgenic are also shown, right. And the relative fold change for this particular, so different lines are also clearly shown, okay. And in the field condition, okay, we went to the field also, okay, we tested for I think so 2 years, 2 consecutive years, okay. So 2015 and the 16, so that time, so we saw that this particular resistance response in the by use of the PSS1 under the promoter 2, you can see all are showing the resistance response. But whereas the wild type Williams 82, the accession is showing the susceptible response, right. And this clearly is showing that upon interrogation of this PSS1, right. So this SDS resistance, so in the transgenic soybean plants under the field conditions are clearly visualized, okay. So also against the P. soja in the previous slide, okay. So in the conclusion, then mutation in the PSS1 actually compromises the NHR against the oomycete, that is the P. soja, and the necrotroph, that is the Fusarium virguliforme, but not against the P. syringae. And the PSS1 was identified to map to lower arm of the chromosome 3, and it is a novel gene whose mechanism of the action during the NHR is really unknown. And the transfer of this PSS1 gene, that is the non-host resistance gene that is identified from the Arabidopsis, so when it is introgressed into this soybean, it provides the broad spectrum resistance against both biotrophic as well as the necrotrophic pathogen. And that's all. And I would also like to acknowledge my mentor when I was really working with my postdoc, and all my colleagues, and the funding agency from IOSABIN, USB. Also, I did some part of the research in here, so funded from the SCRB and the government of Odisha. So I thank the organizer for inviting me to present the work all over again, and I thank you all for your patience listening me too. Okay, and I would like to have some question if I can answer. Thank you.
Thank you, sir. Thank you for your wonderful presentation. So, sir, we have a few questions coming in, and the first one is from Dr. J.P. Rath. The question is, how far the transgenic varieties demonstrate properties in field conditions?
So as you can see, so those field condition data are really shown, right? So in the field itself, right? So by the application way, so as you can see in here in the field, so 2 consecutive years we have seen the resistance response, and they really behave good. Okay, and we also got the US patent on this one, and just recently 2018 we got the patent, when actually it took 5 years for us to get this patent, and we also applied for international patent recently.
Okay, thank you, sir. Thank you for your answer. The next question is again from Dr. J.P. Rath, and it's in slide number 13. What's the standard deviations?
So let me go there.
Yeah.
So these are the replications, right? So of like let's say the S4 for this particular first one. So we have used, so I think so 8 or 7 for independent plants from this particular recombinant inbred lines, and against that, so whatever the infected, the number of the seedlings, okay, or the percentage of the infected seedlings were really calculated across this particular one, okay, and 3 times 3 biological replication actually we did to really delineate about what this particular resistance whatever we are really getting is all correct, or it is really consistent or not, all the time points or all the accessions that we have tested. Otherwise we are really doubting our own result.
Thank you, sir. I hope Dr. JP, that's query is answered. And the next question, it's coming from Raj Kumar Joshi, and his question is, did you find any difference in the disease phenotypes of RILs having only PSS1 expressed and those which have both PSS1 and PEN expression?
Tell me again.
Yeah.
Maybe I can also see. So
did you find any difference in the disease phenotypes of RILs having only PSS1 expression and those which have both PSS1 and PEN1 expression?
No such difference in the expression. So as we just checked those particular RILs and did the PEN1 in the background check, okay. So all were showing the similar response, almost similar response.
Okay. I hope Dr. Rajkumar Joshi's question is answered. And he has another question. Given that soybean improvement using NHR would involve genetic transformation, bringing in regulatory issues, what alternative do you suggest?
So suggestion is like let's say the transgene may not be introgressed directly into the soybean. Rather, what happens is the soybean gene where this particular similarity is all there. So with that can be so gone for CRISPR-Cas, okay, where so we are not introgressing the foreign gene. Rather, so just changing the own gene, we can so have the particular one in place. Okay, and also the point is, so as long as this one is really not causing health damage to the users, like let's say the sweet corn, which is like having the foreign gene, which the invertase or the sucrase, so which actually convert the sucrose into the fructose and the glucose, that is really accepted, right? So and it is not going to hamper the human health, okay. But as long as this particular gene is really going to be activated upon the infection, this can directly be introgressed, but otherwise it can be changed by the CRISPR-Cas, okay, or the original gene from the soybean that can also be changed, okay, in such a way that the active domains, okay, or the one which is really providing the resistance can be so changed too, or some synthetic construct can be put inside. That way, they will say that okay, this is not non-vegetarian or this is not foreign or so. Okay, synthetic construct can also work.
Thank you, sir. Thank you for your answer. Now, the next question is again from Dr. JP Rath, and he's asking, one candidate gene shows variation in DNA band pattern as shown in slide 15 and 16.
So see in here, so we are saying this one is clear, like tightly showing, so matching to NGA707. So also the CIW20, because this NGA707 is just in between these two one, in this between these two marker like CIW20 and the CIW22. So we say that this is more or less linked to NGA707, keeping in fact the CIW20 and 22, so both in place. Okay, so to actually fine tune where exactly this particular gene is really matching to or mapping to, we go for the fine mapping and then found out is really so placed in between, so with the region 2.69 megabase pair. And so before that one or the across the two border markers, so there are your heterozygous plants. Okay.
Thank you, sir. We have another question that's coming from A. Ratan Kumar Singh, and his question is, resistance biosynthesis pathway or reaction will be same when a particular host is attacked by a necrotroph and biotroph in same place, same time and space.
So that is quite possible because infection is infection. The plant does not like plant, although they know that okay, it is a necrotroph for the biotroph, but the infection is infection. Okay, so but against the infection, from the plant side, so some sort of the resistance response must be coming up. Okay, in the format, finally that will really try to defend itself. Okay, by expressing the certain like those reactive oxygen species or the different set of the reactive oxygen species. Those are the actual bombing agent or against those pathogens. So it is quite possible the similar pathway might be also activated. That's why I'm showing like let's say this PSS1, how the gene is really functioning still we do not know, right? Although we just found out that upon deployment into the soybean, it is providing resistance and the localization is onto the plasma membrane. Okay, might be it is doing something. Okay, so maybe it is providing some sort of the resistance, activating the others or the downstream set of the your activators, which really kill the pathogen. It is quite possible. So it may be the same.
Okay, thank you, sir. I hope Ratan Kumar's query is answered. And we have another question that's from Mohammed Haris. Are genes responsible will be same against all kinds of pathogens?
No. So our genes, though these are like really expressed against the cognate partners. Cognate means which really it can recognize too, right? And so those are very specific, very specific against the specific group of the adapted pathogen. But non-adapted pathogen, they really cannot do the business. Okay, so they really are not against all kind of the pathogens. But the NHR genes, yes, they are.
So the next question is from Dr. Kumar, and the question is, NHR is polygenic in nature. Is there any monogenic controlled NHR?
So NHR is polygenic, yes, it is true. But like in our case, when only single gene was really mutated, we see the breach of the so resistance was there, and clearly so multiple cell death entry was there. Okay, but we are still not saying that okay, this is the only gene which is really providing resistance. But in addition, maybe some other background genes are there which really, but maybe this PSS1 gene that we identified is the key regulator in the pathway. But also, so to further explain this one, so from our group actually, so we have 30 independent mutants. Okay, and recently we found out 5 more NHR genes, and they are under review stage, and someone is in I think so. Recently we got the acceptance from Plant Journal. Okay, so that is a PSS30 gene that is identified, and that is localizing to another chromosome, that is chromosome 5 lower R. Okay, so basically there are many genes which might be also providing resistance against the P. sojai, and so also against the other pathogen it might be working.
Thank you, sir. I hope Dr. Kumar's query is answered here. And the next question is again from Dr. JP Rath, and he's asking how many base pair mutations in PSS1 gene you have marked.
So it is only single gene mutation. Okay, I think so. I have shown that one in this slide. Okay, I have not shown it, but it is a single point mutation where actually it is a non-synonymous mutation. Okay, so it was converted to aspartic acid. Okay, so glycine residue was changed to aspartic acid at the active region. We did not go for the modeling and all. Okay, maybe we are trying to really check that one, but it is a single point mutation.
Thank you, sir. So next question is again from Dr. Andi Kumar, and he's asking PTI seems to play it if the non-host and host are taxonomically distant apart. ETI seems to play if the non-host and host are taxonomically closer. What is your comment?
So yes, so PTI, as you said, so in the non-host and host are really taxonomically distant, but ETI, so for the non-host and the host as is really found out is like in the ETI. So that time the factor really upon the recognition by the R molecule or the R molecule from the plant side, so might be really working if they are really closer. Okay, that's why the R gene, as pointed out, so they really provide the resistance against the adapted pathogen to some extent or for one generation, but upon exposure to the plant might not provide durable resistance. And as for the host and the non-host are concerned, so the ETI by the deployment of the non-host resistance gene, so may be providing broader spectrum resistance and which will be really also durable. Also, recently we have checked those PSS genes that we identified. Also, those are showing response against the rice pathogens. Those are also communicated very recently to the journal and they are under review.
Thank you, sir. So the next question is again from Dr. JP Rath, and he wants to know NGS 707 DNA band variation is marked.
Yes, so you can see so that DNA variation in the sense, so you are saying like whether the molecular mapping position is only shown, but with this one actually the genomic sequence we are not associated. So here only we are just using this particular information for the mapping only to just see whether this is in a homozygous condition or not.
Thank you, sir. The next question is from Dr. Andi Kumar. So he's asking, soybean and Arabidopsis are taxonomically distant from each other. Can we expect single gene control NHR here? What is the difference between immune? Oh, I think that's the next question.
Yes.
Please take the first
question.
Yeah.
So no problem. So actually, as we have seen, that the transfer of the Arabidopsis gene in the soybean is really providing resistance, although they are taxonomically distant from each other, or they are really distant by evolutionary trend. Okay, maybe they have evolved or they diverged 200 million years ago, but transfer of the Arabidopsis gene is really providing the resistance, and so as we can see that this is really not showing complete resistance, but still some background susceptibility response is still there, right? So still those susceptibility responses are there, but might be so some sort of the resistance is really not fully active. Like let's say in the wild type, as you can see, we have also one type of the plant which is having the R gene incorporated, or the MN1606, where the R gene is introgressed in that particular line, is showing complete resistance. Okay, but whereas the generated transgenics by introgression of the PSS1 is showing resistance or improved resistance, but not to complete 100%.
So we have another question, and that is again from Dr. Rondi Kumar, and he's asking, what is the difference between immunity and NHR?
So immunity in this sense, like the total plant immunity, right? But NHR, so they provide the resistance. So starting from like use of the gene from distantly related plant species, which is a non-host, and the immunity, as you say, the plant's own immunity. But this will be saying, like let's say the NHR, the Arabidopsis is is providing immunity against the Pisozyte. Okay, but immunity as a term, so it will be explained like from the host side, right? Whatever the power is there, it will try to resist. That is the immunity power, or the innate immunity of the plant, as we say. Okay. And like let's say the host plant, so they are susceptible to this particular type of the infection, but the non-host plant, they provide the immunity utilizing the non-host resistance gene. Okay, like let's say people are finding so many of the resistance gene. I think so I was listing 47 number of the NHR genes so far have been identified, and few of them are deployed in the crop and tested in the field condition. They are providing actual resistance. Okay, so from the non-host plant side, the resistance is provided, that is non-host resistance, and from the host side as well as from the non-host side, so the whatever the resistance is provided will be actually called as the immunity. And as per the terminology by the Jones and et al, as their common reference or the understanding, this immunity can be really explained.
Thank you, sir. Thank you for the answers, the elaborate answers
to
these questions. And so are there any more questions coming in? We have a compliment from Faryad Khan. Excellent use of visuals in the presentation and great presentation, he's saying.
Thank you.
Any more questions? Okay, we have a question coming from Ram Prasanna, and he's asking, can NHR induce immunity?
So induction of immunity in this sense, so it is not actually induction, rather its involvement in the immunity upon the introgression, so it will involve itself to activate the defense. Okay.
Thank you, sir. I hope his query is answered. Any more questions? Okay, sir, I think we are done with the questionnaire now.
Okay.
Thank you so much for your talk. It was really enlightening. Yeah. Thank you. So I think we can go ahead with the next speaker. Optimizing NGS data analysis plays a vital role in genomics. And to make it easier, we have genome stations to rescue. And to know how it works, we have with us Mr. Avinash Dhar. He's a genomics product manager at Bioinnovate. He specializes in bioinformatics and has been a part of Bioinnovate for the past 4 years. He has trained close to 1,000 students, research scholars, scientists, sorry. So may I invite Mr. Avinash Dhar to deliver the speech? Sir, sorry, excuse. We have a last minute question coming in for Dr. Vinod. So do we have you with us?
Yes.
So the question is, in case of BT cotton, it has showed resistance to bollworms. but showed the susceptible for minor pest whitefly. Why can't happen same in transgenic soybean?
So it's like the plant which was really generated in the like BT cotton. So that actually showed the susceptibility because the resistance whatever is provided. So are they just first instance, but later on the pathogen or the insect they develop the resistance or their power to really overpower that particular gene, right? Then they just kill the plant and just eat upon. Okay, but this soybean transgenic, if at all they are there, so actually we tested, so that they are providing resistance against the two different type of the pathogen. One is the hemibiotrophic, another is the necrotrophic pathogen. So they are actually broad spectrum. Okay, and as the Arabidopsis and the soybean, they diverged 200 million years back. Okay, so as per the orthologous gene and characterization, the homology actually is less than 40%. Okay, so we believe that this will be really providing so broad spectrum, and it is actually whatever the like let's say the mutation is really taking place in the pathogen may not so be subverting this immunity provided by this PSS1, that is the non-host resistance gene from the Arabidopsis. So yes, this will be providing so broad spectrum and might be also durable. Whatever the change the pathogen might be incurring, right? So still the resistance will be provided, but not the R genes. But the R genes which are so not durable to that extent. But for few certain select pathogen, but to some generation they can provide the immunity.
Thank you, sir. Thank you for the last minute query, Pooja, and thank you, sir, for answering the question.
Right.
Now I would like to invite Mr. Avinash Dhar to take over the stage and share with us his presentation.
A very warm good afternoon to everyone. I hope my screen is visible and my audio is fine.
Yes, please go ahead.
Okay, thank you. So, to begin with, I'd like to thank IPS for providing us with this opportunity of co-hosting the webinar and including one of our sessions in the same. Also, a big thanks to all the speakers who very graciously agreed to participate in the webinar and share their insightful presentations with all of us. Now, what I'm going to talk about in my session is an optimized NGS data analysis approach, uh, the genome station experience. Now, before we talk about what exactly a genome station is and what does this quote unquote genome station experience stands for, we start with few basics which I think all of us know as of now. So in June 2000, a press conference was held in the White House, which kind of announced an extraordinary feat, the completion of draft of the human genome, which later got published in the year 2003. So, like, why this event was such a big landmark? The whole reason was it was for the very first time when researchers were able to read the 3 billion chemical letters that make up the human DNA molecule. This was for the very first time that something was a scientific experiment was carried out at such a big stage with participation of researchers from different countries and a huge budget attached to it. And during the progression of this experiment, there were several genomes that got sequenced alongside. Uh, Arabidopsis genome, the mouse genome, to name a few. So, since then, since the completion of this human genome project, the progress that has been made in this particular domain of sequencing is phenomenal. The cost of sequencing has reduced drastically. When we talk about the experiment as such, it took us $1 to sequence a single base, and now we are talking about sequencing an entire human genome in under $1,000. And the same has been reflected in sequencing the genomes and transcriptomes of other species as well. Now, what has this led to? What impact has this caused in the overall scenario as such? Now, this has led to DNA sequencing becoming an everyday tool in life science research, and like it has more or less improved upon the outcomes quite drastically. Now, if we talk about data in general, we know for sure that this 21st century is the age of big data, correct? Every now and then we come across ads which constantly keep on reminding us that data is the new oil, our decisions are data driven, whatever we do is governed by the data that is surrounding us, right? Genomics, for that matter, is one of the largest sources of data generation in the entire world. And if stats are to be believed, then it's about time genomics takes over YouTube and Twitter as the single largest source of data generation in the entire world. So you can very well imagine the amount of data which is being churned out through various genomics based experiments on a daily basis. Now, it is also said that by the year 2025, the rate of sequencing will be almost 1 zettaBase, 1 trillion base pairs per sequence per year. So this is the amount of bases that we are sequences that we are expecting to generate in the coming future. And if we talk with respect to the human genome, then we are anywhere estimating to sequence around 100 million to 2 billion human genomes by 2025. And if we combine all this genome sequencing that is going on around us, there will be more sequenced genomes than there are humans on this planet Earth. When I talk about sequenced genomes, I'm talking about all species in general. So we are currently flooded with data coming out of various genomics experiments running all across the globe. And this has caused what I have titled this slide as, it has caused the genomics data deluge, which is the flood of genomics data. The genomics data is flooding our ecosystem like anything. Now, before we talk further, generally when we talk about once the data is generated, obviously the data in itself is inconsequential till the time we derive something meaningful out of it. And the meaningful information depends from researcher to researcher, as was very beautifully explained by Dr. Sungu in his first slide, that the data itself doesn't hold that much information. What you want to achieve through it and how you are going to process that data to achieve your research goals is something which holds more importance. So most of the times whenever we generate our data, the approaches that we use to analyze the data are governed by a single factor, that is presence or absence of a reference. So based on this criteria, we can divide our sequencing approaches into two categories. First one is a de novo sequencing approach, uh, which is characterized by the absence of a reference, genomic reference to be precise. Second is a resequencing approach, which is characterized by the presence or availability of a well characterized genomic reference. Now, some commonly worked upon applications in the field of genomics include whole genome sequencing, transcriptome sequencing, targeted resequencing, genotyping by sequencing, chip sequencing, metagenome sequencing, small RNA sequencing, and bisulfite sequencing, with each application targeting or focusing on specific applications. Like if we talk about studying gene expression, we opt for transcriptome sequencing. If we talk about studying just the coding regions in a genome, we'll go for targeted resequencing. If we want to study DNA protein interactions, we'll go for chip sequencing. If we want to identify the microbial content which is present in a biological sample, we go for metagenome sequencing, so on and so forth. If we want to study methylation effects, then we'll go and epigenetic studies, we'll go for bisulfite sequencing, so on and so forth. So these are some of the commonly worked upon genomics application in the research sector. Now, Uh, if we talk about tools that are available, well, the list is endless. Even during the various sessions that were conducted over the 2 days, our esteemed speakers covered various tools that they had utilized in analyzing their respective data sets. So if we talk about last year, between 2020 2021, close to 325 new tools for various genomics data analysis based applications have been published all around the globe. uh, and these include tools catering to both alignment as well as assembly related functionalities. So we have improved genome alignment tools like Sibelia Z, Callisto, Salmon, to name a few. We have interesting algorithms like pseudo alignment coming into picture. Then we have sophisticated genome assembly tools like Vengen, WTD, BG2, and the Shasta toolkit. And these are just a few significant ones which have found quite an acceptance amongst the research community. Uh, the list is ever increasing and is endless. And every now and then we look at new and much more sophisticated tools coming out and trying to replace the existing pipelines and protocols on one aspect or the other, be it uh, a computational uh, be it their computational poweresses or the less load that they are generating through intermediate files which are being generated through any analytical process. Every now and then your tools come up with one plus point to replace the previously existing tools and pipelines from the market. So it becomes absolutely necessary for all the researchers and everyone involved with analyzing and handling genomics data to upgrade and update their analysis pipelines as well. I mean, because sticking to the same pipeline over and over again, although it might be a faithful pipeline which has served you for quite a long time, but yeah, there comes a time when even publications demand you to use a certain set of tools and a certain pipeline that is up to date, that is abreast with the current standards out there. And the biggest plus point about most of these tools is that most of them are open source and are freely available. You can download them, you can modify them if you are into coding, and yeah, you can modify them to serve your purpose or optimize them any which ways. But when we talk about all the plus points here, there are few negatives involved as well. As they say, uh, there's nothing which is completely black or white. There are shades of gray involved everywhere. So yeah, same is the case with this flooding of genomics data and analytics based tools which are available to us. So the biggest problem here is how to handle the data. When I talk about handle, it includes storing it as well as processing it to derive meaningful information from it. So if we talk about the sequencer in general, if we consider the current scenario, your sequencers are not producing complete genomes that, if I have to give an example, you can read writing a newspaper. It will give you output in the form of shards or pieces of paper that you have to join together and form or construct something meaningful out of them. So if we talk about arranging the pieces of a jigsaw puzzle together, that without a reference maybe, or even with a reference, sometimes it ends up becoming a very complex task even at this stage when cognitively we are far more advanced than what we were in our younger days. Similarly, if I compare the same situation to building Lego blocks and building something, constructive out of it, it involves a lot of time, effort, and patience. Now that is a limited stack. Maybe the biggest of the Lego sets would constitute of around 10-20,000 pieces, not more than that. Maybe say at the max a lakh pieces that you need to assemble to form something meaningful. But here, when we talk about genomics data, the reads, as we call them, the bare minimum, the basic unit of any sequencing experiment, the reads that come out of your sequencer, the stack is so large that you cannot deal with it manually. So you require tools that will process them. And those tools don't usually work on your regular systems. You require computationally superior systems to install and utilize those tools to their maximum potential. A very small example would be, um, all of us would have tried opening Word documents in our systems. So a small 10, 20, 30 KB file or a 100 KB 1 MB file opens quite easily. The moment we start to open a heavier file, say a 50 MB dissertation thesis of ours, which includes lots of pictures and text based content, there is a slight lag in our system. Now imagine a file which is 100 times bigger than that particular dissertation thesis of ours. Obviously it will be difficult for our system to process that data alongside, right? So yeah, a need of specialized computational system or a computational architecture will always be there. So apart from this, as I said, handling of genomics data does not only deal with analyzing and interpreting it, it also involves storing the data somewhere. uh, there is strictly a need for us to move ahead from the conventional data storage sources that we use: CDs, pen drives, DVDs, hard drives, external hard drives. Now we need to switch over to more sophisticated storage media that can help us to secure and keep a backup of our data on a regular basis. Now, the irony here is that no matter how much we stress about the importance of sequencing and about the importance of utilizing up-to-date computational architecture for analyzing the huge amount of data that is being generated, uh, the sad fact is, as sequencing machines improve and appear in more and more laboratories, or if I put it in a way, key sequencing becomes more and more accessible, the same cannot be said about the condition of state of the art computational and storage infrastructure that is available throughout the country. So there are several reasons which are responsible for this problem. So most of us are a bit skeptical when it comes to setting up a computational infrastructure at our lab or in our facility, and the reasons are quite genuine. The first and the biggest reason is the inability to figure out the optimal configuration for the establishment. You might have a budget in hand. Most of the times, you might end up going overboard, or you might end up procuring a system that is underwhelming, that does not cater to your research needs based on your sample load, right? That is one of the most common scenarios that we have observed, and we can attribute it as a reason for this skepticism. Next is the fear of command line. Although there are a lot of commercially available pipelines around us, but one of the biggest reasons, uh, like a, they are quite rigid, and b, the licensing costs are quite expensive. So most of the tools which are accepted in publications as such are based out of command line. So and the operating system involved in general is a Linux or Unix based operating system. Ubuntu, CentOS, you might have heard a lot about them. So that is a big challenge, because as biologists, we tend to be a bit wary of the command line, the black screen and the blinking cursor. We tend to be a bit wary of it, right? And the third and most important thing is, who is going to put the facility to use? That is another factor which is contributing to this skepticism quite a bit. So now, as I said previously, the commercially available toolkits for data analysis are expensive, rigid, and do not cover for the computer hardware. You might end up procuring a toolkit or a workbench which is available commercially, but it does not cater to your system requirements, correct? You If you are, say, many of them come with a recommended computer configuration, so you end up paying for the license separately, and then if you don't have that configuration, you end up upgrading for the same separately. Two different expenses, proving out to be quite a handful for us if we are running short of funds, right? So the solution, let us discuss what solution we as a service provider in this domain can propose you guys here. So Before we talk about the solution aspect of it, I'd like to introduce you to Bionavid. Established in the year 2011, we are an ISO 9001:2015 certified company. And if we talk about our offerings, these are the various verticals we deal with. We started off with analytics, offering a state of the art and publication friendly analysis services pertaining to genomics data to the researchers all across the country. And over a period of 10 years now, we have worked with premier research institutes all across the country. And we had the honor of collaborating with quite a few speakers as well from this webinar as well, and a lot of members in the audience. Uh, During our journey of 10 years, we have been able to generate close to 80 plus publications in this domain, and numerous acknowledgements, and that itself is a testament to our acumen in this data analysis and sequencing domain. Next in line is designing of various software solutions for genomics data analysis. We have automated platforms like SQIT, a workbench for dealing with genomics data. So you don't have to worry about the command line. You just have to fill a short form, press on submit, and your analysis is done. We have a specialized cloud storage and project management platform called GStack, wherein you can upload your data, store it securely. You can either opt for a remote installation, which is at our end, or you can opt for a local installation as well. If you want to host a copy of GStack in your institute, you can do that. So that gives you a two-way data security. You can store your data locally as well as on the cloud. Also, it is a project management platform wherein you can keep a track of all your data and the stages in which your project is going on currently. Then we have the genome education vertical, which I am also actively associated with. The genome education vertical deals with spreading awareness about the domain as such. And we have been conducting various programs for students, research scholars, and research scientists. And we have over the past few years conducted quite a few workshops on various genomics data analysis based applications. If I specifically talk about the 2020 lockdown period, we have been able to train more than, I think, 1,500 research scholars from all across the globe, including areas like Czech Republic, Peru, apart from the regular hub of genomics activities like the United States, Singapore, United Kingdom, and other European countries. So yeah, now let me come to the genome stations. These genome stations, I'll talk about them in detail in our coming slides. These genome stations basically are specialized hardware solutions that we have developed specifically to cater to NGS data analysis. How do they do that? I'll explain it in the coming slides. Now, this is what we have proposed, and this is what we are this is what we are presenting here in this presentation as well. So as a solution to all the problems that were discussed in the previous slides, Bionavid proposes a proprietary product which we have developed in collaboration with Dell. What does it boast of? What are its features? It's an optimized hardware for better performance, 20% enhanced while handling genomics data. It boasts of a customized operating system that comes pre-installed with the latest open source analytics tools, a graphical user interface based analysis package SQIT to simplify NGS data analysis, simplified automated pipelines for analyzing NGS data, and access to a hybrid and dedicated genomics cloud storage platform GStack. So this is what we have managed to bundle up together for all the researchers out there in a package that we like to call as the Genome Station. So we have variants here from desktop to server level solutions. Depending on your budget, we have a Genome Station to suit your need and your requirement. So this is the white paper that Dell had published on its website, which states how Genome Station improve and enhance your experience of handling and working on genomics big data. This Dell covered as a case study on their website. Now, what is the Bionavid advantage here? Because why, like the question here is, why should you opt for a genome station? Apart from all the features that were mentioned in the previous slide, wherein we talked about uh, how it's an optimized hardware solution and how uh, y- the it comes with the SQIT package, it comes with various automated pipelines, it comes with various open source tools installed. uh, we provide complete hardware and software support for your genome station. So most of the times when you procure a system, a regular system, not a genome station, a regular system, maybe a HP or a regular Dell system, uh, you are just covered for the hardware. All the installations that you end up doing are on your own and at your own risk, right? You don't get any support for it, and moreover your device does not come in as a plug and play solution. Once you procure the system, you spend another month and a half to make it ready for your genomics data analysis experiments, right? But this genome station, since it comes pre-installed with tools that you require, we also cater to software related issues if there are any in your system. So any issues with the operating system or any new tool pertaining to genomics and proteomics, rather omics in general, if you want to install, we'll provide you complete support for that alongside the regular hardware related support. Also, we have, as I said previously, we have inculcated various automated solutions for all your analytics requirements, right from raw data quality control, as in when you get your data from a vendor or when you get it sequenced, right from step one till the last step, which may be, say, getting a gene network or annotation for your gene network for various differentially expressed genes, or annotation for your assembled genome or a circular genome. Everything will be covered through these automated pipelines. Any updates released over a period of 3 years will be plugged into your genome station at no extra charges. Also, with over 9 years of experience in the domain of genomics, what we are running into our 10th year. What we also ensure is that any support or consultation that you require to best utilize your genome station, team Bionovid is up for it. Obviously, we won't be doing the entire analysis for you, but just to give you a blueprint of how to utilize the system to its fullest capabilities, uh, with respect to the experiment that you have in mind, we will most surely help you with that. Again, this genome station comes with options of online on-site training, online being the new normal, but if required and as and when the conditions improve, we also provide trainings. That is kind of a mandate. It comes along with the package as such. Also, in case if you require skilled manpower, and people who are expert at analyzing genomics big data and are accustomed at handling it in a high performance computational facility, we can help you source those manpower as well. So that is the Bionavid advantage, the advantage that you get when you procure a genome station. Now if I have to show to you about SQIT, which is our flagship analytics platform, this picture on your left is a command which shows, you know, uh, which deals with quality control of your NGS raw data. Now, if you look at this command carefully, it runs into 3 lines and there are several parameters involved with it. Okay, those who would have run these tools on command line would know that a lot of parameters are associated with any command that you are, any tool that you are looking to run, be it something as simple as performing the QC to something as complex as assembling a genome, right? So it will involve a lot of parameters, and someone who is new to the entire experience of command line, it becomes difficult for them to acclimatize themselves to such an environment, right? And typing such big commands on the terminal. Now replace this, keep this in mind, and replace this interface with something like this. Here, the same parameters have been translated into simple forms, right? The same command has been translated into a simple form with each parameter displayed as an input point, right? All you have to do is just write down your desirable values in the space provided, load your samples, and click on submit. The moment you click on submit, you are good to go. Your analysis is automated, and at the end you get all your filtered files along with customized reports, which will have inputs based on our experience regarding the various aspects of your data, right? So this is the workflow configuration. So we simply fill this form, and once we click on submit, provide our input files, click on submit, we can get a detailed report which covers various aspects of analysis, and it is loaded with various figures and representations and graphs, uh, heat map, gene network being some of the commonly used ones. Again, we have with us what I want to talk about next is what we call as GStack. So GStack is a dedicated cloud platform which also doubles up as a project tracking platform. Now, regular cloud storage looks something like this, wherein you can simply dump in your data, and it is not specific to genomics in general. Means the same drive can be utilized for different purposes, and there's no way you can track the usage depending on the applications that you are working on, right? Now imagine this, and now look at this interface. I'll show you a demo video of the same as well. Here you have a dedicated interface wherein you can keep a track of your project, how much space you have ended up using, what are the various applications you are working on, how much data each of those applications is consuming out of your allocated storage. And you can add collaborators, share your data, load the data. I mean, it gives you a comprehensive it GStack is a comprehensive platform wherein you can manage, store, and track the everything related to your data, right? So it becomes quite easy for researchers who are looking to move away from your regular data storage equipments, I would say, like pen drive, hard drive, right? This is a very hassle free way of transferring, sharing, and managing your data. Now, if I have to give you a short demo of the same, if you could excuse me for a minute. Yeah. So I'll be showing you a brief demo of these products as such, both SQIT as well as the GStack interface. So this is how you start SQIT. We have a file here called the genome station version 1.jar. All you have to do is just type this command, press enter, and click on this link. Copy it in a web browser and just open the link The moment the link is opened, this is your interface. You can
Avinash,
yes sir.
sorry to disturb you. I think your this screen is not visible. It's the same slide which you were
Oh,
presenting
is it
earlier.
just a second sir.
Yes.
Is the video visible now? Yeah,
Yes.
I'll just start over. Thank you for telling this sir. Yeah, so this is the SQIT interface. All you have to do is open the terminal, and as we speak here, this platform has become this package has is now platform independent, and we are slowly working on reducing this dependency of typing this particular command java-jar genome station.jar. We are working on getting rid of this dependency as well, and making a icon for it which will just open normally like you do in Windows. Like you just double click on that icon and the interface opens up. We are almost in process of developing something similar. So this URL, as I said previously as well, the moment you run this command, you get this URL. All you have to do is copy this URL, paste it on your web browser. You're led to this interface. Now if you register, if you are a new user and you want to register yourself here, you can do that. It is hosted locally, so even if internet connection is not there, you can access this tool without any problems. And if you want, you can host it over a static IP, and the entire institute or entire setup can access it remotely as well. So yeah, so once you register, enter all your credentials here, set a password, click on sign up, and you're good to go. Now post login, this is how your interface will look like. Once you register, you are expected to log in with the details which you had used while registering. So you log in, and this is your interface. This is your username here on the top right, and the various applications that you can work upon. The list is growing. This was a previous version for which I have recorded a video. The list is ever increasing. Now we have uh, GUI based interfaces for bacterial whole genome analysis, exome sequencing data analysis, so on and so forth. So yeah, once we log in, suppose we are looking to perform QC for our data, we just have to click on, if we are working on Illumina data, we have to click on short read. Just we have to fill this basic form with all the details specific to our institute. If required, we have to set the parameters here. By default, we have set few values here. If you are looking to alter those values, you can do that. Otherwise, if you wish to go with the default values, you can do that as well. And if you want more information about the parameters that have been included in this form, all you have to do is take your cursor and bring it to this question mark symbol, and it will give you a detailed explanation of each and every parameter that you are about to enter a value for, right? So yeah, once you add your samples, you click on submit, you get this message here. Just click outside anywhere and you will have access to your tool again. You can go to the current and history tab, look for instances. It will show that your QC is running. So once the process is complete, status may it will show that it is done, and next step will lead you to what you can do next with your data. So once the QC is done, obviously the next step would be a detailed report wherein you get an idea of what exactly is there in your data, correct? How is the quality of your data, right? So this is how that will look like. So you just have to fill another form here, a short form with all the details that you want to be printed on the report. Select the output folder, and this is the report that you get, and it will have all the information regarding the quality of your data. And as parting note, we have added our opinion about the data as well, as you can see here. So overall data quality is very good with 82.67%. High quality reads have average read length of 63 base pairs, and these reads will be considered for further downstream analysis. So this is how the SQIT looks like and functions. Obviously there are updates which are happening on a regular basis, and we are working on it. Next up is GStack, which as I said was the demo, which I said was your genomics cloud and data management platform. So yeah, if you look at the interface, you can access it anywhere through internet. You just have to enter your user ID and password here. Once you enter your user ID and password, this is how your interface looks like. So it tells you various projects which are going on, what is the size each project is occupying, how much data was available and how much you have ended up using, what stage your analysis is into. This is again a very old project that we had, and what stage your analysis is into and how many stages are left further. So all of this can be managed quite easily by you. Also, if you are looking to add data and collaborate, you can do it here. So this is the data storing and data download interface here. So yeah, this is pretty much about GStack. You can add access data from here and download the raw data files quite easily. And if you want to add collaborators, you can do the same through this interface as well. So this was about GStack. Now if we talk about the various installations that we have managed to set up for these genome stations, ranging from, as I said and as I had shown in the previous slides as well, ranging from desktop level units to big server level installations. We have been able to set these systems up in institutes like JNU, ICGB, DU North Campus, IBSD, Tamil Nadu Agricultural University, TMC Kolkata, and a lot of other institutes, both private as well as publicly funded. And the count goes in excess of 100 individual units. And if we talk about the clientele, they are not restricted to a specific domain. We have clients working on varied applications ranging from small RNA seq data analytics to eukaryotic whole genome sequencing. And if you talk about the organism spectrum that the clientele is working on, it ranges from simple prokaryotes to complex eukaryotic genomes. And this is the heat map we are of various genome station installations that we have managed to put up, and this is increasing as we speak. So yeah, I have shown you the demo here. What I would like to do is I would like to share a quick feedback from one of our clients. This installation was done in DU North Campus in Dr. Shailendra Goyal's lab, and this is one of his students who has been utilizing this system for the past year and a half. Let's hear what she has to say about the same. Was the video audible? Just a second.
No.
University of Delhi, Department of Botany, working as a research scholar. The broad area of research in our lab are molecular genetics, crop improvement, and genome organization. As you all know, this kind of work generates huge amount of NGS data, and for the analysis of same, we procure this small but very powerful instrument from Bionavid Technology Private Limited in 2020. Array of tasks it performs are pretty very impressive, from small tasks like local blast to the big tasks like complete transcriptome assembly and DEZ discovery are very successfully performed by using this. Big genome data for comparative analysis were also achieved by this instrument. Also, as a user, I personally feel that this is a good addition to our lab, which had saved a lot of time because it had its pre-installed softwares and packages which are most commonly used in our research area.
So this was Dr. Shailendra Goyal's student talking about how Genome Station has helped in making their day-to-day analysis related work easy. Right. So if we look at an ideal analytics facility, this is how the structure would look like. Uh, we are talking about a centralized computational facility, which would be a server, ideally comprising of uh, ideally comp- ideally comprising of a centralized HPC, which is a high performing computational system, uh, say around 96 cores, anywhere ranging from 38 to 96 cores with around 2 TB of RAM and 12 TB of internal storage. This particular setup would be interconnected to individual nodes comprising of 64 GB RAM and i7 12-thread processors, which would suffice for your daily analytics needs like basic bioinformatics or 2 to 4 samples ka data analysis. Then to back up all the data, a network assisted storage and uh, should be attached, which again is backed up to GStack, which is our hybrid cloud system, and you should have a centralized power backup facility for your setup as well. Now this is an entire, when we talk about setting up an infrastructure, this is what an ideal infrastructure should look like. Things vary based on your budget and requirement, and yeah, individually also these work fine if you're looking to sustain a lab, or if you're looking to sustain a single lab. So yes, you can do that as well, but if you are looking to set it up uh, at an institute level, then obviously this is what should be what you should be looking at. Finally, if I have to talk about an ideal processor and RAM combination, well, most of the times, as I said in the beginning as well, if you are uh, about to procure a computational system, the biggest thought block that you end up facing is what configuration should I go for. So if you have any resequencing based experiment and you're looking to handle 2 to 4 samples at a time, then you can go for a 32 GB RAM, 4 core 8 thread setup. If you're looking to handle 4 to 8 samples, then you should look for a minimum of 64 GB RAM with a 6 core 12 thread setup. And if you're looking to handle 8 samples and beyond, then you should look for a minimum of 128 GB RAM, 12 core 24 thread setup. Similarly, if your sample load and your samples are coming from a de novo based experiment, then your computational requirements tend to jump up a bit. So if you are looking to handle 2 samples, then again 32 GB RAM should be enough. If you are looking to handle up to 6 samples, a maximum of 6 samples, then 64 GB RAM should be sufficient. But if your sample load increases anywhere above 6 samples, it goes 6 samples and beyond, then minimum 128 GB RAM 12 core setup is what you should be looking at, right? It totally depends. Generally, when we talk about de novo experiments, then they are more RAM intensive, and if we talk about resequencing based experiments, then they require more processing power. So you can design your system based on these inputs. So that was pretty much it. I would like to thank all of you for your attention and the time you invested in this presentation. In case if you have any queries, you can drop us a mail at info@bioinnovate.com, or you can call us at 9535 619191. I would request the moderator to drop the mail ID as well as the contact number in the chat as well, right? In case once the situation, the entire situation normalizes, please feel free to pay us a visit. Our office is in Kasturi Nagar, Bangalore, and the exact address is mentioned here, right? So thank you so much. Now I'm open for queries. Um, if you have any questions, you can drop them in the chat section. Okay, so Dr. Bhaskar Chandra Sahu is asking, in Odisha, where is your genome station? Uh, the one of the major installations that we had done was in Dr. Lambodar Behra's lab, as Mr. Rohit has also mentioned in the chat, NRRI Cuttack. And is your genome station installed in any institute near Ranchi? Uh,
Hello, ma'am.
yes,
Hello,
yes, sir. Yes, sir.
ma'am.
Hello.
मैं ये बोल रहा था मैम आपको कि
Dr. Sushil, I think you are not on mute. So we have installations. I mean, not exactly in Ranchi, but in Calcutta, we have a genome station installed in Cryzaf. Dr. Deep Narayan Saha, if I'm not wrong, his installation, we have an installation there. What is the processor required for such analysis? It depends, sir, totally depends on the sample load that you are about to subject your system to. As I had mentioned in my previous slide as well, if and the experiment that you are looking at. So yes, sir. So if you are interested in procuring a system, I mean, I have noted down your name. If you could drop your mail ID and contact number in the chat, we'll be more than happy to have a detailed discussion with you. I have taken a note of your mail ID, sir. Our team will get back to you, sir, with all the detailed explanations regarding the same. So if you are looking for a system, so as I said previously, there are a lot of commercial toolkits available as well. Sir, it depends. As I said, the minimum computational requirement depends on the experiment that you are performing. So if you are, I'll refer to my previous slide again. If you are looking for, if you talk about our supplying capabilities, then we can supply systems ranging from 32 GB RAM to maximum of, you know, sky is the limit for that. It totally depends on your budget. But if we talk about sample load, minimum 2 to 4 samples in a resequencing experiments, as I had discussed previously, wherein there is a reference genome involved, then you will require a minimum of 32 GB RAM and 4 cores system. And if you are looking for de novo sample load, de novo experiments, 2 to 3 samples, you can go for the same configuration. The moment your sample load increases, the overall configuration also changes, right? So I have noted down your query, sir. GPU is available as per request. CPU, as I said already, the 4 core 8 thread part covers the CPU, the number of cores that would be arranged. And sir, the costing information and all, we have few promotional offers running as well. The detailed detailed list I would share over mail to you, so that you can have a look at it, and we can further understand what exactly are your requirements. We'll be sending a detailed mail to you for the same. And yes, Dr. Rahul, I have noted your mail ID as well. We'll get in touch with you. We'll get in touch with you. Just a second. Yeah, yes, sir. Medusa is a standard genome finisher which is used for reference-based genome which we use it for for genome finishing. Yes, you can use Medusa. It is a standard one, and moreover, it is freely available online, so you can utilize it. It will be a bit, Dr. Basavaraj, it will vary a bit as far as the combination here mostly deals with the whole genome part. It would vary if you are looking for transcriptome data analysis, specifically for transcriptome data analysis. I have taken the upper limit here. If you are specifically looking for transcriptome data analysis and that will be your only application, then it would vary a bit. As far as Ratan Kumar sir's query is concerned, I would like to refer it to Dr. Awadh. If you could drop in your mail ID, I would request Dr. Awadh to get in touch with you and he will give you a price code for the same. Any more queries?
Avinash, hello. Avinash, are you there?
Hello. Yeah. So, sir, as far as your queries regarding Dr. Andy Kumar, as far as your queries regarding the these wet lab aspects, as in DNA isolation for PacBio and the NBA rules are concerned, I might not be the right person for it. We'll get back to you through mail in case if you're. Like, I might not be able to answer it because I'm not the right person for it. We'll get in touch with you through email, and I would request the concerned team members to get in touch with you. Any more queries specific to the system as such, or any analysis protocol? If you are interested, you can please drop them in the chat.
Yeah. Sushil, with your permission, I would like to, rather than putting in the chat box, because
Yes,
a lot
sir.
of pausing moments are going on.
Yes, sir.
To Avinash, it was a very nice, enlightening, basic information about genome sequencing. In fact, we have been doing this, the training part in IRI funded by NHP COST for the last 2-3 years, the genomic assisted crop improvement and management. Under that, we precisely involved several companies to deliver a talk and conduct a practical session. I really appreciate IPS and the persons who are connected to this event in different places of our country for creating some kind of awareness among pathologists, young pathologists and students about the importance of the subject genomics. I really appreciate. So there are a couple of things coming to my mind. Number one is I already put something in the box. This information need to go down to all the the scientists, the rules and regulations, what is being practiced in our country. For example, many times genomic DNA sharing it with the company, as long as the machine is present in the country, there is absolutely no problems. But the problem comes whenever the company says they may be having their principal elsewhere, maybe located in Europe or maybe in Korea, maybe in China or some other country. This is not really clear. It is not really clear to me either. So what regulation? This is very important, because on one side we talk about National Biodiversity Authority, who says none of the biological material from our territory can go out of the country without their approval. So on other side, we are freely exchanging the material for any kind of intended genomic purpose. So where we draw a line. So this is one of the very important point. Everyone should know what this I discussed with many companies also. As Avinash clearly told, none of the companies really came out with a practical solution for this. So all we presume the machine is available in India, and with that presumption, we are sending the sample for library preparation and sequencing. So this is going on, and I'm sure many people who are involved in this kind of work would really endorse these views. There must be some kind of clarity, especially from the company side, because when they enter into rate contract with the academic institution, this point need to be discussed as one of the terms and conditions, so that the clients are not having any doubts in their mind before sharing the biological material with anybody for any purpose. This is very important. So I really appeal to the industry, because it is not the if you say that we need to take permission, then it's a separate channel altogether. We cannot do any kind of genomic. It's very difficult. So there must be some kind of some guidelines. This can be discussed at the time of rate contract entry into different institutions. This is one point. Avinash, any take on that?
Sir, actually, if I talk about my views on it, obviously, we here, would definitely be following all the prescribed protocols for the same. But as I said previously, I might not be the right person for it, because I don't deal with that aspect. Dr. Awad is there, who is handling all of this. So what I would request everyone is, in fact, what I would say here is, I would ask Dr. Awad to get in touch with you through mail, and he would be able to give you a better insight about it, because my domain specifically deals with the systems and informatics part of it. As far as the protocols involved in sample collection and what protocols are followed once, if you have to outsource the entire sequencing process as such, that is Dr. Awad's domain. So I would request him to get in touch with you over mail.
Yeah, perfect. Yeah, let us from the regulation to something core basic. I have some couple of questions. You installed different genomic workstation in different places in the country. I really appreciate you. The question here is the software program, whatever you are installing in that that workstation, is your own proprietary one, or it is from public open source one?
So, sir, what we have, the entire system is a proprietary product. It has been developed in collaboration with Dell, and we call it as Genome Station. And the reason why it is a proprietary product is because our team has, collaborated with the Dell R&D team to optimize the overall performance of the system while handling NGS data. As far as the tools are concerned, what we have observed based on our experiences, and I think, sir, you would also be adding some valuable information in it. You have, as I heard in your talk yesterday, that you have used CLC Genomics Workbench. As far as though many times having a rigid approach to doing analysis might not suit the bill every time. You need to mix and match your tools based on the requirements, right? So we prefer to have open source tools installed in our system. Now, the drawback with having open source tools installed in a system, specifically if it is going to be handled by a biologist, is the fact that most of us might not be that confident with the command line, because these open source tools come with a Linux background, correct?
True.
So what we have done is, what we have spent our internal R&D is on writing various scripts to optimize the performance of these open source tools, and then embed them in a GUI based interface. Like, it would be a graphical user interface, wherein instead of you typing the command on the terminal, filling on all the parameters with the recommended values, we'll give you a platform wherein all you have to do is fill out a form. The recommended values for each of the parameter would be already there. Based on our experience, we have already set some default values. If you want to continue your analysis with that, you can click on submit and you're good to go. Otherwise, you can alter those values. So this is what we have improvised as far as the system is concerned. So we are providing you with the flexibility of an open source tool, plus in case if you want to utilize a simple GUI based interface, then we are inculcating that as well. Now,
Okay.
how it is different from any other commercially available pipeline is that pipeline does not cater for your system configuration, isn't it? Now, this genome station is an entire package. So you get the pipeline plus the hardware at the same price. You're not paying anything extra for it. So that is the biggest advantage that these systems have.
Okay. So this workstation is both hardware and program come in a bundle, correct?
In a bundle, and we install the software based on your need and requirement.
Need and it's not a one standalone for everything. It may not be like that, correct? It may be customized for every application.
Yes, exactly. That is why we have launched this product. We wanted to serve each and every individual based on their need, their research requirements.
Okay, great. So I do not know, can we have some kind of commercial discussion here? It may not be possible because many audience might be curious to know what would be the total cost. You need not tell the precise one. Maybe it varies from customer to customer, but nevertheless, you can put a range. What would be the kind of initial investment if somebody wants to do it? If that information is shared with us, it will be very useful. At least for somebody like me who can, call you again after some time.
So we are actually, we would be more than happy to assist. In fact,
suppose see, most of us, 1 second, most of us, you put different kind of options here, depends upon the sample size. Suppose the beginner, maybe 2 to 4 sample. I think for 2 to 4 sample only going to be the kind of hot selling in future, because nobody will have a high end. They will be having their own internal needs. So you can tell what would be the pricing or what will be the range of price for 2 to 4 sample, the 8 thread and
Yeah. So
hello, Avinash.
32 GB RAM, 4 cores.
Sorry for the interruption. I think Dr. Awadh is online. Hello.
Yeah. Hello. Yeah, sir. I'm Dr. Awadh. Yeah. For the 2 to 4 samples for 32 GB, approximately it will be of around 1.75 lakh.
1.75
lakh.
1.75 lakh, including hardware. Huh?
Yeah, including hardware, software, and our support for the bioinformatics.
Okay. So 32 GB RAM and maybe 4 cores. What would be the storage? 2 TB will give, or how do you storage? Maybe unlimited or cloud?
Sir, inside the machine you will have a 2 TB hard drive, and based on, so you also get an additional I think around 300 GB of cloud storage for 6 months, 6 months to 12 months, and there is option of taking a paid subscription of the GStack Cloud as well, and you can upgrade it later.
Okay. Yeah, it's okay. It's a very competitive price only, whatever you are telling. The cloud storage, suppose for some reason we are not able to sustain our project in the long run, still we'll be having access to the cloud?
So for the subscription period, yes. And once the period is over, we would request you to kindly download the data and keep a local backup of the same in your system.
Okay, so the cloud storage, as long as a subscription is there, you can enjoy. Otherwise, you need to transfer it to hard drive. Yeah.
There would be some grace period involved as well, wherein you can download the data and keep a storage of it. But yeah, it will mostly be subscription. Mostly is subscription based. Yes.
Okay, great, very nice. And another most important point is the government system of taking the service from private players. You need to enter into a kind of rate contract. I'm not sure in how many institutes you are in rate contract. So that is the first step before I choose your company for any kind of work. Not me, I'm telling, I'm talking on behalf of everybody here. So rate contract assumes a very important point here. So keep in your mind. So in IRI, I think you know it very well. Only 3 companies are in rate contract. That was very surprising to all of us. And likewise, I think you need to enter into a kind of rate contract within every institute because you are promoting genomic science among all the students. I think to make it real in their lab, I think you need to do that. That's my first, yeah.
That is a very good comment, sir. And we have started working on that. And we are applying as many as rate contract as possible.
Yeah.
And mostly we are trying
there
to
is no way anybody can choose any company. Yeah, that's
true, sir. Yes, sir. True, sir. Also, we are trying for GEM also for this product. Soon we'll complete that procedure in like next 1 or 1.5 months. So that option will be also there by next financial year that you can directly purchase this product from GEM also.
Yeah, the machine hardware is from Dell only. It's not from some local one, correct? It's a
No,
branded
sir, it will be a branded one, Dell. They are our OEM partner,
Okay.
and all the like warranty, hardware warranty is being covered by Dell.
So you have manufacturer's authorization also for you, right?
Yeah, yes, sir.
Okay.
Actually, we have white paper. We have worked with the Dell R&D team, and we have like optimized the hardware with the Dell R&D team as per the NGS bioinformatic data analysis.
Okay, fine.
Yeah.
Okay, I put a question on Medusa. One small question. When you do a 50 to 100 MB genome, I think most of the pathologists, they handle pathogens and their size is in the range only. So maybe 0 to 1 to 100 I'll put. Suppose the genome size increases, then the chances of the gaps are more, correct? So the gaps we are filling through Medusa program, how reliable it is? Is it audible?
Sir, could you please repeat the question?
I think there was a network break. What I was asking, when you are using Medusa to finish the genome, obviously you are going for reference-based assembly, correct?
Right.
Yeah, yeah. So the gaps are filled with the help of the so-called reference one.
Correct. Yes.
Is it really reliable?
You're talking about Medusa as an approach.
Yeah. Suppose now recently I come to know that somebody has assembled in a single contig. I was so surprised. So then I asked, then they told we have used kind of that paper appeared in nucleic acid research, I believe, the Medusa paper. So they told we used a Medusa program to fill the genome and finally they got the single contig. So how reliable and what is its acceptability in the international forum? Suppose if you are submitting a paper, will it be accepted?
Hello. Hello, Dr. Kumar.
Yeah, hello.
This is Dr. Roli here.
Yeah.
So I would like to answer that question.
Please.
Whatever you have asked.
Yeah,
so
please. Thank you.
Medusa, actually, Medusa is used for definitely, as you said, that it is a reference-based approach. But what we are doing, why we need the reference is we just want to anchor whatever contigs or scaffolds we have made. We just want to anchor it based on the reference. And if there is a little gap which is needed to be filled, it will be filled so that you can get a maybe pseudo chromosome at the end.
Yeah.
And the leftover contigs, whatever.
Yeah.
So this is a very, well known approach, and it has been used in a lot of papers. Medusa basically it is used basically for prokaryotes, but eukaryotes there is another tool called Ragu, which is used. So basically it is very well like taken approach. I mean, you can find it in publications and all.
Okay. So now the nanopore has come. They are promising that single molecule sequencing, and the PacBio sequel 2 is also there. So where do we stand now? Suppose if those prices are going to be cheaper, then probably I would go for single molecule sequencing only. Why I should use Medusa and still putting some sequences, pseudo sequences, which may not be the real. So what is the projection from the company?
So basically what you are saying that you can make a genome out of using the nanopore instead of going for and I don't think that is possible, because the thing is when you are using the single cell this nanopore sequencing, it has lot of errors. So because of that, it is not feasible to make a genome. So that is why we always prefer, if you have a bigger organism like some eukaryotes and you might want to make an assembly out of it, you should go ahead with an hybrid approach. That will be little cheaper compared to not only using the longer reads. Longer reads you can use to make a good scaffold, and then shorter reads you can make error correction. Yeah. So that is the approach which is being followed. And now with the high C reads and all, more correction methodology is there, so you could end up getting a good assembly at the end.
Yeah, great. Okay, thank you. That's all I wanted to interact with all of you. And I once again thank IPS and the people who connected. Sushil is anchoring this program and so many others. And I really appreciate, and we need more and more this kind of program in future as well. So we'll make it a kind of regular affair. Still, genomic is established as a kind of laminar flow in every pathology lab. So let us do that. Okay, thank you.
Thank you.
Yeah.
Sir, thank you very much, Dr. Ondi, sir, for very elaborative and nice discussion. We all are benefited with this. Sir has raised very pertinent point with regard to this NBA guidelines. Actually, we are also not very clear, and we really need to see like if a sample is to be sequenced outside the country, can means DNA of course may not be shared, and if after library preparation it can be shared after fragmenting the nucleic acid and preparing the library. I don't know that maybe sir, maybe in many committees he may take up this. And but as sir has said, in a rate contract, while entering to the rate contract, it has to be very clearly said that this genetic material will be sequenced in the country only by any private company. So thank you, Avinash.
Thank you.
Now, thank you, sir.
Thank you. Yeah.
So just like this brings us to the end of the webinar. The past 2 days have been filled with insightful talks from renowned researchers who presented a very crisp overview of how omics-based techniques in general are playing an important role in understanding host pathogen interactions and plant metabolic pathways in general. As far as today's session is concerned, while Dr. Chungu's talk stressed on the use of genomics and proteomics-based techniques in gaining a better insight into understanding specialized metabolic pathways in plants, Dr. Sahu through his talk uh, emphasized about non-host resistance in plants. We also had a session from Bionavid, wherein the role of a specialized computational setup in handling NGS was highlighted. Now to present a vote of thanks, I would invite Dr. Sushil Sharma. To give concluding. Sorry for this. To give concluding remarks.
Avinash, you can this unshare the slides.
Yeah, I will.
Then we'll go for concluding remarks from Dr. MS Gurjar.
Yeah. So as Dr. Sharma said, I would like to invite Dr. MS Gurjar to address the webinar and give concluding remarks for the same. Excuse me, sir, you are on mute, I think.
Okay.
Yeah.
Dear participants, today we have finished today's webinar on NGS to deciphering the host pathogen interactions. Concludingly, we can say that it's successfully conducted by the Indian Phytable Society and Bionavid, and all the speakers who have delivered nicely all the topics, especially genomics and proteomics part, to understand the pathogen and host interactions. Because NGS is a bigger tool which we can understand the pathogens. The pathogens which are diverse in nature, also fungi, bacteria, viruses, which are need to understand. Until unless we go for sequencing, then we can understand the each isolate and strain also. Even you can see nowadays the COVID virus is also different strains are evolving. So that's the NGS technology is very much useful to understand the host pathogen interactions. So finally, I can congratulate all to Bionavid and our society secretary Dr. Robin Gogoi and joint secretary Dr. Kalyan K. Mandal, and from Bionavid Dr. Rohit Sukla and Dr. Avhad, and a special thank to Dr. Sushil Kumar Sharma who continuously conducted all the sessions very nicely, and hope all the participants will understand, get benefited these all talks, and it is very much useful. And I would like to thank all executive members and all IPS members of the society who have given the support attending this 2 days webinar, and it will be very much useful in research work. So thank you one and all.
Thank you, sir. Thank you for your kind words of encouragement. And now I would like to invite Dr. Sushil Sharma to address the vote of thanks.
Very good afternoon, Very good afternoon to the participants and dignitaries who are attending this webinar since last 2 days. So since now we have reached to the final conclusion of this 2 days open webinar, which was on next generation sequencing for deciphering the host pathogen interaction, which is organized by Indian Phytopathological Society and BioNivit together. So may I take this opportunity to thank the IPS Secretariat, Dr. P.K. Chakravarti, President IPS, Dr. Robin Gogoi, Secretary IPS, Dr. Kalyan K. Mandal, Joint Secretary, and Dr. Malkhan Singh Gurjar, the Treasurer, EC members, and particularly Mr. Rajkumar sir from IPS for all help and immediately accepting our proposal to organize this webinar in very short span of time. I would also like to thank all the lead speakers who had been delivering very nice and informative talks since last 2 days. I would like to thank Dr. Professor V.K. Barnwal, Dr. Ondi Kumar, Dr. Divya Chandran, then Dr. Professor N.K. Churungu, Dr. Sahu, Mr. Avinash for their informative talks since last 2 days. Finally, I would also like to thank all the participants for their patient hearing and very nice interaction during this webinar and participating in this event. I will not be doing justice if I don't thank BioNivit team for their initiative to organize this open webinar, which actually has helped us to really refresh our knowledge and also learn like how this NGS can further revolutionize the plant pathological research. So from BioNivit, I would thank Dr. Rohit Shukla, Dr. Awadh, then Dr. Roli, Mr. Avinash, and Madam Kranti for coordinating the webinar and co-hosting it. So thank you one and all. Thank you.
Thank you one and all. Thank you very much.
Thank you, sir. Thank you for your kind words. The certificates will be emailed to all of the participants, and I would suggest that allow us a time of 5 working days, and please do check in the spam folder also. There are many times that the mail lands in the spam folder. You can check and you can mark us not spam, so that next time if you get our any mails from us, you can see them in your inbox. Thank you for your participation. Thank you one and all.
Thank you everyone.
Thank you. Thank you. So now we can end the meeting.
Yeah.
Yes, sir.